Good morning, good afternoon, good evening, wherever you are, and welcome to this session on WHO consultation on novel manufacturing technologies for monoclonal antibody production with a focus on prevention of infectious diseases for LMICs. This session is recorded. We will share the presentations as well as the links to the link to the recording. During this session, please don't hesitate to ask your questions in the question and answer Q&A box. The questions will be answered, I think, if we have time, but we will probably have time because yesterday it was very well organized in terms of timing. So yes, please ask your questions in the Q&A box. And if you have any technical issues during the session, please write in the chat. I will help you as much as I can. Thank you very much, and I wish you all a pleasant session. Erin, the floor is yours.
Thank you very much, Alex. So welcome to day 2 of this WHO meeting. I'm just going to provide a very brief recap and some takeaways, some key takeaways from the meeting yesterday. So what we heard yesterday was about some of the normative work that we're doing here at WHO in terms of the development of preferred product characteristics and some of the regulatory guidance that's being developed or will be developed soon. Basil gave us a really comprehensive overview of how monoclonal antibodies are produced, which I think really highlighted the complexities of these products and why they are so much more costly to produce compared to chemically synthesized drugs. We heard about efforts that IARBI is undertaking to advance affordable monoclonal antibodies, and in particular the work that they are leading on HIV monoclonals. In 2020, there were 410,000 new HIV infections. So clearly we need more tools in the prevention toolbox. We also saw some really promising data from the malaria monoclonal antibodies that are under development. And I'm really looking forward to seeing the efficacy results from that adult study in Mali that Bob mentioned yesterday that should be published within the next few weeks. You also heard about RSV long-acting monoclonal antibodies, and that probably next year these products are going to, or the first product will be licensed, and that there will be other products to follow. And this is for prevention of RSV in infants, where development of vaccines has been ongoing since the 1960s. Without yet a successful candidate authorized. You also heard about rabies monoclonal antibodies and how we do now have 3 products licensed, and these really do address an unmet need where there was a shortage in especially the equine rabies immunoglobulin, which is production technology from over 100 years ago. We heard from Mass Biologics as well about their diphtheria monoclonal antibody that they've got under development. And I think what was important there is that because of the drop in immunization coverage on a global scale, it is very likely that in the coming years, we are going to see increased outbreaks of diphtheria. And so, unfortunately, the equine diphtheria antitoxin is in such short supply. So having something else there to be able to treat patients is going to be really important. And for a while there, it looked like that product was going to struggle through making it to the market because of difficulty in trying to enroll the adequate numbers to demonstrate efficacy in clinical trials. But it seems that there is an alternative pathway forward with the US FDA through the animal rule. We also heard from CEPI about how they see these products and the role that they might have in potentially epidemic pathogens like Nipah and Lassa, chikungunya, MERS and Ebola. And more specifically, that they're planning some work around the advancement of Nipah monoclonal antibodies. And I think throughout the day, we were reminded again and again that these products are expensive to produce. The current production, at least, is very expensive. And when they're given at high doses, not only does this mean the costs increase, it also means that the administration must be intravenous. And so ways to bring down the technology, to bring down the cost through novel technologies or large-scale production, or trying to optimize product attributes so that you don't need such a high volume to be injected, I think is really important. And that's really the purpose of today's session. And we ended the day with a bit of a poll just to get your thoughts on some questions that we had. And the first question we asked participants was whether or not you believed it would be cost effective to use monoclonal antibodies to prevent infectious diseases. And the vast majority of you said it depends. Nobody said no, so that's good. But I must say I agree with the majority of people that I think it's going to depend on the use case. It's going to depend on the pathogen and what that target population is. Because if large doses are required, it's going to mean that the products are going to be expensive. We also asked you which pathogens you thought monoclonal antibody development would help to address an unmet public health need. And you'll see that those pathogens that are in very large text are the ones that were repeated multiple times. And so malaria stands out, RSV, HIV, Nipah, rabies, Lassa, influenza. I see dengue and chikungunya fairly big. And then there's also some other pathogens that were mentioned, as well as pathogen areas as well. We also asked you about what kind of attributes would be needed to facilitate the use of these products in developing countries. Most of you tended to strongly agree that the products should be administered via intramuscular or subcutaneous injection. Most of you were sort of towards the side of disagreeing that IV administration would be acceptable. Most of you sort of at least agreed, maybe not strongly, that product prices must be similar to vaccines, and quite similar that most of you agreed that some of you agreed that products are likely to be more expensive than vaccines. With regards to cold chain, it was towards the agreement that the cold chain requirements should be similar to vaccines, so 2 to 8 degrees Celsius. But then some of you also agreed that products should be able to be stored outside of that cold chain for periods of time. We also asked you whether or not you thought that the development of WHO generic preferred product characteristics would be possible and useful. And more of you thought that these are actually pathogen specific, so it's probably quite challenging to develop generic preferred product characteristics, but some of you did see that there might be value there. So on the WHO side, we'll have sort of a think about that a little bit more, about whether or not we see feasibility and value in developing more generic product profiles. And then so for the session today, I think I already mentioned this, but we're really going to focus on some of the current technologies and novel technologies that are hopefully going to bring down the cost of monoclonal antibody production. This is by no means a comprehensive representation of all the technologies out there, nor does it mean that we endorse these technologies that are presented today over other technologies that are not going to be presented. It's just that, you know, we could only have so many presentations in a virtual meeting. And what we do hope to present is when we do report the findings of the meeting, that we will actually have a more of a comprehensive landscape analysis of all the technologies that are under development. So after we've had those presentations, we're going to hear about from the Bill and Melinda Gates Foundation about what they're working on. We'll have some experience shared from the FDA. And then we'll finish with a panel discussion about what the public health community, developers, regulators, and funders can do to ensure affordable monoclonal antibodies are advanced. And so the final thing that I just wanted to note is that it is a packed agenda. We've got a lot of presentations. Likely some presentations are going to go over time. So we might not have a lot of time for Q&A. But please do pop your questions in the Q&A. If we can't address them during the meeting, we'll try and get the questions answered via email. And if any of you have any thoughts that you want to share via email, please feel free to send me an email in the coming days about, you know, what your thoughts are or what your comments are. So with that, I'm going to hand it over to our first presenter of the day, and that's Wei Chang from WuXi Biologics, who's going to present on their optimized CHO cell manufacturing line. Over to you, please, Wei Chang. Thank you.
Thank you, Ellen. Okay. You can see my slides. Okay, and first of all, thank you for the invitation. What I would like to talk today is really talk about the speed and also touch a little bit the cost for CMC development timeline for anti-COVID-19 neutralization antibody and other biologics. This work are based on the CHO cells. So just a disclaimer because WuXi Biologics is a public company. So I will briefly introduce what the WuXi Biologics does, then really talk about accelerating biologics development. So WuXi Biologics, as many of you know, we are a contract research development and manufacturing organization, which is CRDMO. In fact, we use our platform to support on a yearly basis 150 INDs and 12 BLA. And right now we support 14 commercial projects and use 260,000 liter capacity. And total, we actually enable our partner worldwide to develop 534 different biologics, which is about 20% of biologics on development globally. And the company have globally about 13,000 people worldwide. So our platform is really use mammalian cells to support the development of monoclonal antibody bispecific to ADC, and then also use the mammalian cell platform supporting the viral-based vaccine development. Then use microbial to do the recombinant protein and some vaccine development. And so we actually support companies worldwide from discovery, goes through developability assessment, preclinical development, and then clinical and commercial. And right now, worldwide, we actually have 262,000 liter in operation, and going to in few years doubles the capacity up to close to 600,000 liter. So the capacity and they are based on in China as well as globally in Germany and Ireland, Denmark, and the US. And right now we are going to build an integrated R&D and manufacturing center in Singapore with a capacity of 120,000 liter. So all this capacity we use to enable our partner to develop manufacturing biologics. So out of 534 products which we help our client to develop, and 287 is in preclinical stage, somewhere from DNA to IND, then about 200 in early phase 1, phase 2, and 29 in phase 3, and 14 products we support conventionally right now. So with this platform, with all the experience we gained for last 10 years or so, we have accelerated the biologics development, particularly during the pandemic time. So during pandemic time, we actually enable more than 20 different partners develop both COVID-19 neutralization antibody and viral vaccine. For COVID-19 neutralization antibody, we actually help manufacture 5 conventional and monoclonal antibody. Within less than a year and a half, we made more than 2.5 ton of antibody now used worldwide. And there are several other programs still in the clinical development. In terms of vaccine, we helped manufacture viral vaccine about 100 million doses and of DS, and now used and Europe and other countries. There are also protein and mRNA based on vaccine being developed and manufactured. So the question, especially in response to pandemic, what we have done for last, I would say last 6 or 7 years, is relentless driving the speed from DNA to IND. For typical biologics, and in order to achieve faster in human, we actually reduce timeline from 24 months, and that's 2015, now to about 12 months, pretty much for all the monoclonal antibody by specific right now. So obviously this help company to test those products very quickly. And in addition, for the infectious diseases, we further reduce the timeline and from DNA to IND. And typically we are 12 months for the typical biologics, but like 2018, we helped the company file the IND within 9 months for Zika. Then following year, reduced from 9 months to 7 months for yellow fever. And for COVID-19 antibody 2020, we reduced to 3 to 6 months, which I will spend a little bit more time to tell you how we achieve that 3 to 6 months timeline. Then accelerated the CMC development from DNAs to EUA within 14 months. And so that's a very fast speed. So in order to do that, what we have done is really truncated the typical biologics development timeline from transfection, tox, ecology production, GMP production, and the IND. It's really pushed that to less than half of the timeline. And we actually use 2 different ways to produce material for IND. One we call first generation process, use the batch with a stable pool. In this case, we are able to file IND within less than 3 months, so 2.5 months. Then subsequently, we clone those cells, then use for the cloned master cell bank, and this we call second generation GMP production. In this case, it's about 6 months. So the key, this is all based on child cells, is to use high throughput clone screening, and then also use much shorter cell line stability to enable this timeline. And in addition, to further shorten this timeline, clearly there's a lot of parallel work. And while we doing the pool production, we'll continue cell line development, doing the clone stability and create a master cell bank. And use a lot of high throughput technology, then initiate the process development much early on. So within 6 months, we actually can use cloned the master cell bank to produce material to enable IND. So what I want to emphasize here is that all this accelerated timeline, we don't put a product quality and the product safety at risk. And however, we do accept the business risk because we put a lot of effort to do parallel development. So in terms of the technology, clearly we, based on the childcare 1 cells, have a very robust whole cell library and use proprietary codon and rapid bulk pool, and then use NGS guided clone selection. So in this case, we achieve the speed like 9 to 10 weeks from DNA to master cell bank. And even with such short timeline, we get an average productivity of 6 grams per liter for cell culture. And as I mentioned, we actually ensure quality and haven't seen sequence variants at the genetic level. And this is guided by the NGS. So doing that, this is one of the case study we call, and for the COVID-19 neutralization antibody. And this is an example. We received the DNA literally by email, March 2020, and just after pandemic, and within less than 5 months, we will manufacture the first clinical batch at the 2,000 liter scale. Then first conventional scale, we use single-use reactor, use 6 times 2,000 liter, make the first 12,000 liter batch, and the parallel doing process characterization and then PPQ completion. So within 14 months, and the product received the FDA and the EUA approval. So after EUA approval, we made more than 52 conventional batch and a little bit more than 1.8 ton of the protein delivered to our client and used for 44 countries. And even now, there's more than 16 countries are using this neutralization antibody. And I want In addition to the timeline, which is a very short R&D timeline, 3.5 months, and 14 EUA, I want to mention that within 6 grams per liter of titer, we actually reduce the cost significantly, and to the level which Ellen talked yesterday, and which is roughly at about $100 per gram in terms of manufacturing cost. So right now, I think WuXi Biologics, along with several other companies like Regeneron, like Lilly, and we are unable this timeline, short timeline, 40 months, and from DNA to EUA compared to traditional 10 to 15 years. And so this, we believe this can be used for other biologics as well. And just quickly, in a second case study, we actually also, this is 2 antibody, and we spend a little bit more time from the DNA transfer to us, and then go to the 4 months, which is first IND, and then get the approval. This in this case is a 2 antibody, and get approval in China through a randomized double-blind and placebo-controlled trial. But again, it's a little bit longer than 40 months, but still it's less than 2 years from DNA to approval. So this antibody, this pair antibody right now, and are used in China to combat the COVID-19. So a lot of details are shared in the publication. Particularly, we published this in Reshaping Cell Line Development CMC Strategy in Biotechnology Progress. And last year, we have a first publication and on this approach. Just to summarize, we achieved the average 6 grams per liter, and right now for typical antibody, and 80% of project is 5 grams per liter more. And in terms of product quality, this would be no sequence variance, and we, based on NGS, high throughput NGS, we get accelerated biosafety testing. So this will reduce the timeline, and for pool to 3 months, for clone 5 to 6 months. Then, and for the cost, because high productivity, we actually achieve very competitive cost. So finally, I think within a very short time, I just show you quickly this example and how we accelerate the COVID-19 neutralization antibody development from DNA to EUA. And in this case, particularly, this approach can be used further for combat other infectious diseases, use antibody-based approach. So I just want to thank, in addition to our team at WuXi Biologics and our partner and our collaborator to enable this timeline. So thank you for your attention, and if there are time, I would be happy to answer questions or share any details through our publications. Thank you.
Thank you so much. I have a question which you might not be able to tell us. You said that you can achieve competitive pricing or competitive costs. Are you able to give us a ballpark figure of what your, say, cost per gram is at high scale manufacturing, or is this proprietary information?
I can give you a range. Right now, we are at the cost $100 to $200 per gram. So really depending on the scale. So the cost is less sensitive now to our process and the productivity rather than to how much antibody. So essentially, the volume. The more you manufacture, the lower the cost.
All right, thank you. There are a few questions in the Q&A. So the first question is, do you assess continuous process to produce large amounts of monoclonal antibodies in a short period of time?
Oh, absolutely. I think in this case, because of familiarity of fat batch as well as pretty high titer, for the speed, we haven't used continuous process. But for the typical antibody or bispecific antibody, we have to use the continuous process, push productivity even higher, like to 3 grams per liter per day harvest. And in that case, we actually, in the best case, we are able to reduce cost of goods. I would say in best case to $80 per gram of that level, but not for this program, for some other programs.
Thank you. There's also some questions in the chat as well. How much do you leverage from continuous manufacturing is the first one that I see.
We actually leverage a lot in terms of continuous processing. We have done many antibody like for rare disease, enzyme, and recombinant protein use perfusion culture at a larger scale. And as you know, for rare diseases, some of the indication require very high dose. So we use perfusion culture, then we use so-called direct product capture to enable continuous to the harvest, reduce harvest volume. And as I mentioned earlier, we actually applied these for antibodies as well, then bispecific antibody. And so far, I would say about 25 programs we have are using continuous processing.
Thank you. And just two last questions. There are some more questions, but just in the interest of time. So the next question is, how long does it take you to do viral clearance and obtain regulatory release? And then the other question is, do you use intensive fed batch processing or continuous perfusion?
Yeah, and we actually have the advantage. We have a biosafety testing in-house. So essentially for such a fast program, essentially takes a sample and go to our in-house viral clearance study. And in most cases, it's only take a month to two in order to complete the report. So for this EUA, we have completed the viral clearance study, just like a BLA. So in terms of intensive fed batch and intensive perfusion, we actually use both. For typical stable antibody, we actually use a lot of intensified fed batch. And as you know, when the product is stable, and you actually can prolong the fed batch duration. But for unstable product, like some recombinant protein or enzyme, and we tend to use intensified perfusion to improve the productivity. So both actually are used in various combination, and then we use those and as part of the continuous process as well.
Thank you. And just one final question, because I think it's a good one. It's about where do you see the next phase of innovation in CHO cell manufacturing coming from? And are there more efficiencies that you think could be made? And how low do you think the cost could go?
Yeah, I think this is obviously take a lot of discussion for this question, right? But I will give a very short answer for that. You know, I think we still have some room to push the CHO cell productivity, and but not much. And the major room, I think, came from two aspects. One would be really continuous processing. And in this case, you can further improve the productivity. And I mentioned that we reach 3 grams per liter per day, but I think there's still some to be gained. Maybe can reach 5 grams per liter per day. Then also have a continuous processing for the purification for downstream. Therefore, you have a whole continuous processing from beginning and to the end. If you can do that, and the second aspect would be you actually need a really good PAT, so-called process analytic technology, to enable hopefully real-time release. But I know this is a big topic. If you can enable real-time release and kind of reduce the cost for QC, and I believe you actually can reach at least less than $50, 50 per gram, and maybe somewhere $30 to $40 per gram.
Okay, well, thank you. I think that's some good insight. Okay, well, thank you very much for giving that presentation today. I'll hand it over to the next speaker, which is Crystal Dagoneau from Just Evotec Biologics, who's going to present on optimized antibody production using artificial intelligence and machine learning. And I understand she's got to get to the airport, so we might only have time for a couple of questions for her. Over to you, please, Christelle.
Thank you, Erin, for the invitation. Can you hear me okay?
You're a little bit quiet.
Okay, wait a minute. Is it better?
I think we might just all turn our volumes up on our computer.
Wait a minute time, because I have a few audio options, so I'm changing options. With this one, is it better?
I think so, but I just turned my volume up. Try again.
I think it's fine.
It's fine. Okay, great. Thanks.
It's fine. Okay, perfect. So yeah, so today, good morning, good afternoon, good evening to everyone. I'm going to try to present a bit on how artificial intelligence, machine learning can help optimize antibody development and manufacturing, but I'm also going to speak about continuous processing. So I know there's a lot of interest in that area, and that was kind of missing from the title, but this is well presented, in this deck. So happy to speak about it too. So I'll just pass on the usual disclaimer, because Just and Evotec, actually the mother company, is a public company, so needed to include it. So actually, I like to spend just a couple of minutes on who is Evotec, and I didn't put many slides to describe the company, but just in a few words, this is a public company headquartered in Germany, gathering 4,000 people globally in 16 sites, I think, today. So a lot of them being in the Europe, but also in the US. And what is very original at Evotec is that this company is really bringing a lot of drug development expertise, disease expert, with very innovative technologies and what we call omics, so the data together with really the aim to improve the drug development efficiency, improve clinical success rate, and basically accelerate timelines, but also lower costs when it comes to develop drugs, whether they are small molecules or biologics, because Evotec is really working with all modalities, initially actually more of small molecule companies, but with some acquisition now much more present in the biologic space as well. So I've just put this, one of the slides that usually we present, because I think it really shows that Evotec is acting like a kind of a pharma company, but actually in partnership. So we can really help work from the very early discovery stage with helping identifying the targets or validating them, or they are provided by partners, and we can really support all activities all the way from discovery. So in the case of biologics, antibody discovery, antibody optimization, sequence optimization, through preclinical development, with profiling, etc., or the PKPD, to the CMC aspects, so cell line development, manufacturing, and up to commercial stage. So, and this really in one roof, and again, with the disease expertise, which is usually missing from the traditional CRO CDMO company. And so this is the main originality of the company, but also the type of business model, because we are like a mixed CRO CDMO company, but actually we are signing a lot of co-development programs, even investing in some companies, taking equity. So again, very original also in terms of the type of partnerships we are setting up. So that's the mother company. And actually today we're going to talk about biologics, and this is another Just Evotec Biologics name, which is clearly the biologics arm of Evotec. So it came from an acquisition made back in 2019. So Evotec acquired a company called Just Biotherapeutics, funded by Jim Thomas and a bunch of ex-Amgen veterans at the time. And this, so fully integrated subsidiary. So at Just Evotec, the mission is really to develop and use very innovative technologies to expand global access to biologics. And this speaks very well to this conference and to basically all the challenges that we have to bring biologics to the infectious disease and less developed countries. So the mission is really develop the technologies, have a global access, and really focus also on the affordability of those drugs, because that's a big component, of course, of the accessibility. So I'm going to spend a bit of time on this slide, which is a bit busy, but really summarizing well what we are at Just. So Just has developed a fully integrated technology platform called JDesign, which actually integrates artificial intelligence, machine learning, also continuous processing, and design of modular and innovative facilities. So this can be all integrated to actually develop better molecules and actually accelerate timeline and support all the way from discovery to commercial manufacturing. But all basically components of these platforms can also be accessed independently by our partners. So if I start with what I call the discovery pillar, so actually at Just, but also within the whole Evotech group, we have access to a lot of different technology to discover new biologics. So we are using some more traditional immunization tools, safety display libraries, but also kind of a new technology which is called GL, and which is actually a technology based on AI ML, where we actually use GANs, it's kind of algorithms, if you wish, to generate fully human sequences, which not only show broad efficacy features, but where basically the generation of a library were biased towards bringing more developability into the sequences. So the antibody, not only they are fully human, they are efficacious, they are less, immunogenic, but they will also behave much better when they will be brought into the, CMC and manufacturing steps. So this is, the first example where we apply AI ML to actually generate better sequences. Then, when we work with partners, sometimes they ask us to develop to discover the antibodies, but also they have also sequences readily available, so we can still assess them, validate them, and actually further optimize through another pillar, which is called GMD, so our molecular design platform, which is also based on AI/ML proprietary technology, and we leverage a lot of data accumulated from public data banks, literature, but also with all the data that we generate through our manufacturing teams to actually assess the sequence in terms of developability. And again, making sure that it's fully optimized and that the specific sequence candidate will bring the better titer, purificability, will be easy to formulate, will have long-term stability, the right PK, so that again, when it is brought to manufacturing, we have already de-risked this candidate from let's say, an protein engineering perspective. So then the third pillar of this platform, which is called GPA3 for Process and Product Design platform, it is actually leveraging continuous processing. So at Just, we have, I believe, the most advanced continuous process, because currently we are really capable to do continuous manufacturing, not only for the upstream development steps, but also for the downstream up to the end. So we have really connected all the single unit operations, and we leverage then this continuous approach to also provide very flexible manufacturing solutions, because continuous manufacturing, it brings better quality, better productivity, but actually it also allows to produce either a few kilograms of protein antibodies through 2 metric tons with the same process at the same scale, so same scale of bioreactor, and within the same facility. And then that's the whole beauty of using continuous manufacturing. And then those processes, so basically here we have, proprietary cell lines, but we also use, very well-known cell lines, sometimes cell lines from our partners that we adapt. We have proprietary media to also get to some lower cost operations there. So all those processes, we actually operate them in our very original facilities, which we call J-Pod, which are very modular, very flexible, and cost effective, because again, the way they are designed, we have minimized, let's say, the clean room space, which happens to be the most expensive space, because we use much smaller scale bioreactors and very small unit operations. And because of that, then the cost of building, the cost of operating is actually below what you would expect for a traditional facility. So this is, all part of this platform. And by using this approach, we can really develop better antibody candidates, but also provide more flexible and more cost-effective manufacturing solutions associated to those candidates when they're going to basically go through clinic and get to market. And I've put some example of our partners. So like we are at Evotech, very diverse partnership models, very diverse type of partners, some of them being large industry partners, some of them smaller biotech, some of them non-profit organization, government, and we have very big partnership actually with the Department of Defense, a lot focused on infectious disease, antibody, candidate generation and manufacturing, so in the US. So that's in a nutshell, what we are and what we do. I like to present this slide also because using continuous manufacturing and this whole platform, it's also really helping de-risking the development. And we know when we de-risk, we actually accelerate timeline and we decrease the cost. So we can really de-risk earlier on with this in silico assessment, basically really making sure we put the right sequence in the manufacturing process. We de-risk when we use continuous process because we actually eliminate all the scale-up risk, because again, we use the same process at the same scale. So there's a lot of challenges which are well known, which can be avoided. We de-risk also because we use a lot of automation. Actually, not only this help improve the quality, but also has an impact on the reduction of costs, clearly, because there's fewer FTE, so basically people working to operate the process, because a lot of it is actually automated. And we also reduce the supply, because all our facilities, they are designed the same way, using the same automation, the same equipment, the same scale, and then it's going to be very easy to tech transfer from one site to the other and really have a global supply when needed. And we can do that really easily because our J-Pod facilities, they can be deployed in 18 months. So I know there's been questions about COGS, how we decrease COGS, and clearly when we use continuous manufacturing, we increase the productivity level. So we've been seeing examples where we've been outperforming fed batch productivity by 10 times actually. So clearly we are working in the 2 to 4 grams per liter per day type of productivity level. And the very interesting thing is that we can actually play on the culture duration to adjust to the mass demand. And this is what is shown here on this slide on the right. By just adding 3 days to a continuous process, you can actually add, double the mass which is generated out of the bioreactors. So this example was in a 5-liter scale bioreactor. This slide maybe is even more self-explanatory. Again, we can use the same process, same scale, and you can see that if we just need a few kilograms to do clinical trial phase 1-2, we can just adjust the duration to 9 days. But on the day we will need to actually get to commercial manufacturing, and let's say generate 50 kilogram batch, we just have to adjust the process to 25 days duration. And because we are working with high viability cell lines, this is achievable. So here, this is just a slide on the JPOD, so the facility itself, which is clearly much smaller if you compare to the traditional stainless steel, large facility. So because it's smaller, because it's more flexible, we can really, it's made of pods, which are like manufacturing trains, if you wish. But those pods, they can be added as we deploy a facility. We don't have to basically build all the pods right from day one. So we can actually start with smaller CapEx and increase over time. And we can also reconfigure the pods depending on what we want to do. Change the size of a bioreactor. We can go from 500 liter to 1000 liter. Work with hybrid process, because sometimes we do not connect all steps. It's kind of hybrid continuous, or we can also do fully continuous to make the most out of the process and actually increase the productivity and lower the cost to the maximum. So all of that is actually enabled by those type of facilities. So where we are now, at the beginning, clearly, so just, has been funded back in 2014, acquired by Evotech in 2019. Actually, the first JPOD 1 facility has been set up last year during the pandemic. So it's already up and running, currently doing clinical runs that will be commercial ready in 2 years' time. There's actually the replicate of this JPOD 1 facility being launched in France, in Toulouse. So the first one, this is in Redmond, so in Seattle, US. Second one, actually the groundbreaking ceremony will happen this Friday in Toulouse. And so in less than 2 years' time, we'll have a copy-paste of the JPOD 1 in Europe. And the goal is really to expand this network within Evotech, or with partners, with governments, as the design, the equipment, and the type of automation can only be easily and quickly replicated everywhere in the world. So that's where we are. I just put quickly, an example, because we did also work on COVID-19 antibodies development during the pandemic. So at the same time, in our initial J-plant site, we've been developing two antibodies generated actually in our GL library, started to do clinical manufacturing. At the same time, we were setting up the plant, our first JPOD facility, also in Seattle area. And actually, within 2 years, we've set up the plant, we've developed the COVID-19 antibodies, and in just 4 runs, 5-liter scales, basically we generated more than 30 kilograms of DS material. So that was just a quick example to illustrate how the platform and the JPOD can help with the infectious disease type of project. So more to tell, but short time slot. So happy to take any questions and to extend the dialogue outside of the conference. Thank you.
Thank you so much, Christelle. It's all very cool, actually. And I love the idea of the JPODs. There are some questions. I know you've got to get to the airport, but maybe we'll just do 2 or 3 questions. So there was a question about when the earliest time you would be able to produce phase 3 material using your system. And would that be 2024 or later?
Late 2023 and early 2024. Right now, we are basically having phase 1, phase 2 programs, and we are getting the first JPOD 1 site in Seattle commercial ready for early 2024. So yeah, So all PPQ, all late stage. This is going to be possible next year. And actually, we are already developing some kind of late stage commercial process to be ready to operate them in a year time.
Oh, fantastic. And there's another question about, what's the potential for using this modular approach to expand monoclonal antibody manufacturing, particularly in Africa? Is that something that what would
be that's been actually already discussed sometimes with some partners for Africa and for other regions like that. Clearly, those JPODs, if we are looking at a big demand, because it was I think written somewhere, but I did not mention. Really, out of a full JPOD, we can produce up to 2 tons. Let's say 1-2 tons of antibody material. But we could think of maybe just having 1 pod or 2 pod, just taking, the manufacturing platform, 1 train or 2 train in an existing facility if needed. Maybe we don't need the full JPOD. Maybe the development can be done elsewhere. And then we could think of having even smaller manufacturing suite or facility equipping, sites in Africa or in other countries. So yes, clearly, and that's clearly the goal, to have those flexible modular facility replicated and adjusted to the need in a particular geography or in a particular context, which can be a pandemic.
That's really fantastic to hear. And just one final question. There was a question about the fact that these antibodies are derived from artificial intelligence, and while that might improve the affinity, does this make them more toxic because they're not derived from the human body?
So no, that's not going to make them more toxic, because the goal is really to leverage all the data with the artificial intelligence to make, super efficacious antibody and clearly address the immunogenicity, the toxicity. So this clearly is not going to be an issue, but you're going to face by generating those type of libraries. The affinity, it's a different question. We know that we've been lucky. We've had, already super good affinity for some antibody leads that we've generated with GHEL. For some other example, we know we may need to go through some affinity maturation cycles like we would do with any other technology. So that's going to be a bit antibody dependent.
Okay, thank you very much, Christelle. Bon voyage. You can get.
Thank you, and sorry I can't stay with you, but again, do not hesitate to reach out outside the conference. Just need to get into a flight and the airport this afternoon. Thanks,
Great.
everyone.
Thank you again. I'm now going
Bye-bye.
to hand
Thanks.
the floor over to Mark from Didactic. So Mark Eppelmark will be presenting on their C1 technology, which is a fungal expression system. So the floor is yours, Mark.
Hi, I can't share. There we go. Okay. Can you see my screen?
We can see it. It's not yet in presentation mode.
Yes, okay. Where is the full slide deck?
Yep, it's good now.
Okay, great. First, thank you, Erin, for inviting us. It's an exciting time for the industry as a whole, because it's like Moore's Law, things are moving faster, quicker, cheaper. Although with biotech, it's happening even at a more rapid pace. And at Dyadic, we want to talk about, we've developed a thermophilic filamentous fungal cell line. And you'll see over the last 3 decades that we use industrially to produce massive amounts of proteins to feed, fuel, and heal humankind. So the next slide, somehow it's not advancing. There you go. We're a public company, so this is our forward-looking statement. So we're here really because of access and affordability to the middle and lower income countries. It's not existent. Even in America, if you heard Scott Gottlieb back in 2018, 2% of the American public had access to biologics, and it was 40% of our federal budget. That was before COVID. And 70% of the new drugs are biologics. So this is an unsustainable model, even in the West. So what we're doing is we're trying to transform biomanufacturing. We're not trying to improve it. We're trying to completely change the game. Sort of what Elon Musk has done in the auto industry. But we believe that our prior experience in feeding and fueling the world with companies like Shell Oil, BASF, DuPont, and others is leading to the most effective, rapid way to develop and mass produce proteins that can close the gap in the least developed countries. And this is something that's already happening. It's not something we're thinking about, we're already doing it. We're doing it in Africa, we're doing it in India, and we're discussing with other countries. So the timeline of the development of this technology, just to give a little background, is I started the company in '79, selling pumice stone to wash blue jeans. Enzymes came about in the 80s. We started to develop our own enzyme using the same C1 technology. And by 1994, we were already running 150,000 liter fermenters. At that time, probably about 60 grams per liter. And we took that up to about 150 grams per liter of total protein and 80 grams per liter of a single enzyme purified. We had FDA GRAS approval, which I think in some ways is more difficult than getting an FDA approval in the human pharmaceutical side. And we'll talk about where we're advancing our COVID-19 antigen and antibodies towards that approach. So it's about affordability, it's about complexity, and it's about access. What I mean there about complexity is how efficient can you tech transfer technology to countries in middle and lower income, and where they can develop simple, easy, efficient ways to develop and produce their own products to be sustainable in the long run. I don't believe we can provide technologies from abroad to middle and lower income countries in the long run and make them affordable and sustainable. So one of the things that we have to do with this technology, because we use it industrially for 2.5 decades to make massive amounts of low-cost proteins, is bring it into the regulatory and get the approval. Because everybody's always asked, have you been in humans? The answer is we've been in rabbits, chickens, mice, hamsters, cattle, and all kinds of animals, showed safe, effective, protective, and now we're about to go into humans. We're just waiting any day for hopefully South Africa or SAFRA to prove our first in human clinical trial with a protein produced from the C1 technology. We've completed cGMP manufacturing, toxicology, drug substance, drug product, fill finish. It all went extremely well, extremely safe. And one of the things that differentiates us from E. coli, we don't have endotoxins, we don't have mycotoxins, we don't have viruses like CHO or baculovirus. So in a downstream viral, we can save money and time for time release and purification. Our genetic toolbox is unparalleled. It's getting better, quicker, faster, cheaper, just like mentioned with Moore's Law. Just in the last year alone, and we'll talk about on a monoclonal antibody, we've gone from 2 grams per liter using a certain approach to 11 to 22. And that's in 7 days, not in 14 days. And you'll see the morphology change of the fungal cells led to hyperproductivity, low viscosity, robustness in fermentation process, and large scalability. So we have different approaches. This approach here we used, and we'll get into a proof of concept we did for COVID-19 for both antibodies and antigens, where we can do site-specific integration, which means we can pull out a gene and put the next one in without changing the genealogy very quickly. So within 7 weeks of receiving the gene plasmid or gene synthesis, we can not only have a stable cell line, but we have purified protein that can go into an animal study right away and then further be purified for cGMP production later on. And we can put in multiple copies in different sites. This example here shows you we can put in 2 copies, one are a light and heavy chain on the front end and light and heavy chain in the back end. And we have additional technologies now that can do it in even higher numbers of copies at higher yields. In addition, we can do other things once we have this technology. We've created a stable platform, stable cell line, and we can add different secretion and folding factors and improve the performance even higher. So one of the things we had to do is reduce the protease activities because filamentous fungi are the most prolific producers of protein in nature, but they also produce a lot of proteases. And we removed 15 of the proteases one by one to avoid CRISPR and other technologies backgrounds in this case, to come up with a very low background of proteases, where now we have a stable background host where you can see on the right that led to high productivity and increased stability of antibodies and a whole variety of other proteins. The other thing we did, which is either useful or not, and that's the jury's still out, for infectious disease, when you want to have neutralizing and binding activity, we believe that our natural form, and we're proving that in our non-human primate study, which is more productive, can actually give the results with the safety and efficacy that you need for infectious disease, neutralizing binding antibodies. But if you want to make fully human or human-like, we've glycoengineered C1 to make human proteins with the right glycan forms. And in fact, we can make them with, I think, even higher homology to what you want on a consistent basis. So this is an example of a proof of concept that we did for nivolumab. You may or may not be familiar with nivolumab. Nivolumab is the $8 billion oncology drug for cancer from Bristol Myers Squibb. And the average activity of yield is about 4 grams per liter in 14 days. But you'll see the 14 days is the time it's in the fermenter. It takes much longer than that to get that freezer into the fermenter, and we'll get into that. In this case, we were able in 7 days to demonstrate 22 grams per liter in 7 days of a stable active protein. And we're going to improve that further. We can run in stainless steel bioreactors using fed batch technology at very wide temperature and pH ranges. If you move CHO cell a half a pH to the left or right, you start having issues. We can go from 20 degrees to 45 degrees. We can go from pH 5 to pH 8. And that makes a difference depending on the protein. So if you want to make neutral proteins at 37C, pH 7, we're right there. That's our ideal temperature, just like CHO, but we can move it to the left and right further and farther to achieve consistency on these difficult to express proteins like bispecifics, trispecifics, or antigens. And we've grown up to 500,000 liters. So what we're doing here is we're scaling down. And our media cost is 1/20 to 1/50 the cost of a CHO cell. It's basically synthetic media cGMP with glucose. So this slide depicts not only the time in a fermenter is much shorter, but we can run 3 or 4 batches in the same time you run 1 batch of a CHO cell. Typically from a freezer to the end of the fermenter, you can see here it's 12 to 14 days versus 41 to 54 days for CHO cells. And the reason for that is we have 2.5 hour doubling time instead of 24 hours. So what does that mean? Lower cost, faster development. We can create stable cell lines and protein purified for animal studies in 7 weeks. We can run at de minimis cost on media at higher yields, faster, and reduce the time and the cost and the equipment costs and the size that you need in a manufacturing plant versus CHO cells. And on these ready-to-use plants, just as she just described in the Just Pod, you can make even smaller footprints using C1 because you can produce anywhere from 5 to 10 times higher yield in the same time frame with much lower cost media without any bio burdens like E. coli has with endotoxins or CHO cells have with the viruses. So when it comes to structure, you can see from here, and we have more details and more data, that the structure of the MABs produced from C1 are virtually identical to those in CHO, whether it be the peptides, the intact mass, disulfide bridges, oxidation, deamidation, etc. Here's an example that was done for multiple times. Forget the yield of highly, in this case, 24.5 grams per liter. And by the way, that was done in the old days without the new tools. So we're going to see how far we can push that. But you can see here that the binding is virtually identical to CHO, and the neutralization is identical to CHO as well. And you can see here for Andes virus, Zika, Rift Valley, and SARS-CoV-2, neutralization on the antibodies produced in C1 are virtually identical to CHO. So here's an example of a proof of concept that we've developed in conjunction with the scientists of the ZAPI program in Europe on what's called the G7 Monoclonal Antibody. And this antibody, not only do we produce at C1 rapidly, not only can we produce it in significant amounts, quicker, cheaper, on lower cost media using standard microbial bioreactors, but this particular antibody covered all the variants from Wuhan to Alpha, Beta, Gamma, Delta to Omicron 1 and 2. Now, Omicron 4 and 5, we're working with another company on developing an antibody to make a cocktail. And you can see compared to the Regeneron 10933, it was a far better protein that can produce in large amounts at lower cost. But we're really the enabler of speed and cost and yield. We're not really the enabler of discovering and developing antibodies, but in this particular case, I'm just showing the example. Here you can see, again, whether it's a vaccine or an antibody, we can make recombinant protein antigens as well. We've made Wuhan, Alpha, Beta, Gamma, Delta, Omicron 1 and 2, and Omicron BA5 is in development now. So we can make multivalents that can be produced in large amounts at low cost with a small footprint. And we can do the same thing for monoclonal antibodies, blend different ones. So in this case, we're using the 87G7 as the base and spiking it with the new antibodies that come out for the variant of concern. So again, the platform is comparable to CHO in terms of neutralization, binding, and other properties. We can produce things in less time, stable cell lines, lower media costs, faster, use smaller footprints in terms of manufacturing plants, similar binding and neutralization, but also we can have human-like glycans, and in our animal studies, in a non-human primate, it's showing exactly the same performance with and without the human glycans versus the human-like glycans, or we have fully humanized glycan C1 cell lines. We can also work with other therapeutic proteins. We've worked with dozens of them. Here's an example. Again, prior to the new technology, we got 7.62 grams per liter in 7 days of a bispecific. And here's an example of a variety of different types and complexities of different antigens, antibodies, therapeutic proteins, from influenza, HA, NA. You see here we've developed not only H1N1, now we've also developed H5 and H7. We have a COVID antigen, SRBD, from Alpha, Beta, Gamma, Delta, Omicron, Wuhan, etc. We now have a ferritin, F10, SGRBD, at 3 grams per liter in 5 days, and the MABs and the FC fusions, the FABs, the trispecifics. And we even now have an antigen we produced at 10 grams per liter in 7 days. Tox study, not only did it show on the DY100 antigen, which is going to be the first in human protein we put in a human being, if everything works as expected. We hope to do that sometime, start that trial in October of this year. And you can see here, not only was there extreme high safety, but there was persistence and long-lasting efficacy. And you can read the different articles on site. And to the bottom line in the end, a vaccine or drug that's unavailable or unaffordable is 100% ineffective. And in this case, what I mean is we have a 2.5-hour doubling time versus CHO for 24 hours. Our media costs are de minimis compared to the CHO cells. You can use smaller footprints. And I think the opportunity is endless, and we're just getting started. And I'm open to answer any questions you may have.
Thank you very much, Mark. And I really liked your final comment there, because I agree that if it's not accessible, it's not effective. So there is a question in the chat. Could you speak about the cost per gram using your expression system? And they also missed your comment about the what the FDA said about the C1 platform, and has that platform been used for any licensed products?
Yeah, so our first licensed product is going to be in South Africa, and that's on the COVID-19 booster vaccine from C1 that we're producing at very high levels, very low cost. that I talked about going through CMC, CGMP, drug substance, drug product. So we haven't gone to the FDA yet because of the mRNA and the timelines of what's going on. But I have spoken to many people there, including Peter Marks, who loves the technology, but we didn't get the funding from Warp Speed. So we're not operating at rabbit speed, but we're operating at the tortoise speed, knocking things down one by one, moving along the way. And so on the upstream side of the equation, again, if you can get 20, 10, 15, 30 grams per liter in 7 days versus in the fermenter, 12 to 14 days of 4 to 10 grams per liter with CHO with a much lower media cost, you know, the cost on the upstream side is dramatically different. On the downstream side, we use protein A or different purification methods. So really, other than we don't need viral inactivation, which saves money and time, I think we heard today, anywhere from 1 to 2 months just to get release. If you have no viral burden, that's a huge advantage of getting speed to the market and getting out faster. But we haven't gone to the FDA yet. We intend on doing that. Not quite sure which product. It may be with a COVID-19 multivalent. It may be with an antibody, a combination of antibodies. Could be with influenza. But we can't tell you what the FDA thinks. But I can just tell you they have no problems. You know, their point in conversations with us is have the data, safety and efficacy data, they'll approve your drug or your vaccine. They don't really care what you use, just demonstrate safety and efficacy, and they have to approve it. And I believe they will, and you're seeing that, you know, in the case of a lot of things today.
Yeah, so with all those cost savings compared to the traditional CHO cell manufacturing and the timeline, what do you think you could probably shave off 50% of the costs compared to CHO cell manufacturing, or you just, I mean, it sounds cheaper because just of what you've mentioned, but have you done any back of the envelope kind of cost of goods comparisons?
Well, we did preliminary early on at a 14.5 gram per liter, not 22 gram per liter protein that had aggregates and other things in there. And that was about 30% savings. So I don't think your 50% savings is out of line, and it could go even lower. But it's not just the savings, it's speed to the clinic. It's the smaller footprint, smaller fermenters you can use, they get the same output. Again, we can use stainless steel or single-use bioreactors. So if you want to have in-country, middle, lower income country production, we've already developed the technology. We can out-license it. We've already out-licensed it in India. We've already out-licensed it in South Africa. We've actually in South Africa, they can not only produce proteins, they don't have a facility of their own yet, they're thinking of building one, but they can actually program the cells. We've taught them how to engineer the cells so they can do it on site. And they're working on malaria and HIV and a variety of other things in addition to COVID-19.
Great, thank you, Mark. There's just one final question and then I'll hand it over to the next speaker. So this is from the chat. In terms of culturing conditions, you mentioned the broader temperature range that the system is amenable to. What about other parameters such as aeration, etc.?
Yeah, so the unique morphology of C1 allows you to scale it. I mean, remember, we're running at 500,000 liter scale. So we're scaling down anywhere from 10 to 25 to 50 times, in some cases even 100 times. So the aeration is not a problem from VVM. The feed is more uniform because we have more consistent. We don't have typical filamentous fungi morphology. You saw the picture, I can bring it back up, but it's more of a pellet, what we call them propagules, where you can feed them more uniformly. It led to low viscosity, it led to high productivity, it led to uniform feeding and aeration. So that's not a problem. So I think that solves that issue. And by the way, the tech transfer, I can tell you when we've tech transferred it in 2 cases, one with a toolkit company, one of the largest in the world, they actually got higher yields on their first run than we got. And in India, in one of the places that we're working with, within 2 weeks, they got 80% of the productivity that we hit, including purification. And so it doesn't take long. You just stick a cell line in a FedEx frozen package and you're off to the races.
Thank you. It sounds very promising. So with that, I'll hand it over to the next presenter. So now we're going to hear about Plantibodies. from Julian Ma from St George's at the University of London. Over to you, Julian.
Very much, Ellen, and also thanks for the invitation to speak today. Know when you can see this and hear me okay?
Looks great. Thanks, Julian.
So this is going to be a rather different talk. I'm not a, I don't work for a company, I'm a university academic. And so what I've chosen to do is talk more about the plant platform for manufacturing antibodies and to give a broad description of where we're at. I'm not subscribing to any particular one of the platforms. And as you see, there are a number of options which we have available. So I think the idea of plant antibodies really hit the public mind back in 2014 during the Ebola outbreak in West Africa, when a number of medical personnel were infected with the Ebola virus and came back, both the UK and the USA. And of course, we didn't have any treatments or vaccines at the time. And the monoclonal antibody cocktail called ZMapp was administered to 9 people and saved the lives of 6 of them. And subsequently in 2016, ZMapp went into a randomized controlled trial, unfortunately just at the end of the Ebola outbreak. Subsequently, of course, other monoclonal antibodies have proved to be superior to ZMapp. But in 2014 to 2016, at least, the plant antibody concept sort of, I think, firmly hit the headlines, and people became much more aware of what we were doing. This wasn't actually the first time that plant-made antibodies had been trialed in humans. There was an early study back in 1998, and then more recently in 2015, 2 studies here. And the importance about this first study was that it was performed in Europe, where regulatory approval and the GMP manufacturing license, of course, is a prerequisite for going into phase 1 clinical trials. And so a GMP license was issued for the first time for the process of manufacturing an IgG monoclonal antibody in plants. Now, I want to impress upon you that this is not a single technology, but is a suite of biotechnologies. It's become clear that you can use many species, if not most plant species, to produce recombinant proteins, and some of the sort of key examples are here. But in the case of monoclonal antibodies, the 2 key technologies that we're talking about both use varieties of tobacco, either using stable nuclear transgenic tobacco plants, as was used in that 2015 clinical trial, or using a second technology involving transient expression in a variety of tobacco, which is not smoking tobacco at all. In fact, it originates as a weed in northwest Australian deserts. Now, the particular benefits that plants offer, we believe, are several. And cost benefits seems like a no-brainer, but it's probably not in the cost of goods, but more in the possibility of a low-cost early investment into manufacturing. Because, of course, the facilities for growing plants are much less sophisticated than you need for the stainless steel for cell fermentation, for example. And that in itself allows for more participants and more risk-taking, I think, in product development. There's the potential for very large-scale production. And in the past, we've spoken about agricultural scale production. But actually, the productivity is so good now, particularly in the transient expression systems, that going out into the open field probably is not required anymore. The transient expression system also offers extraordinary speed of delivery. And in the case of rapid response biologics, we've really seen some important developments here, particularly in the case of flu vaccines, for example. The thing I'm going to talk about at the end of my talk is the possibility of producing some complex molecules, particularly multimeric proteins, which have been very difficult to express so far. And alongside that, of course, there is, because plant cells are eukaryotic cells, there is very accurate post-translational processing. And finally, I think another benefit is that potentially this is a very simple technology and provides a technology transfer route for biologics manufacturing in low and middle income countries. So, of course, antibodies made in plants are not identical to those made in humans. The protein backbone is identical. The key difference is a subtle difference in the N-glycosylation of IgG antibodies, which is depicted here on the right side of the screen. That was realized very early on, of course, and what that has done is allowed for some very sophisticated glycoengineering to be developed, initially to remove the, let's say, plant-specific enzymes for adding the xylose and fucose residues, which are not normally found in mammalian glycoproteins, and then adding in human enzymes to create a more human-like glycosylation pathway. And as with the last speaker, we now have the possibility of producing antibodies with human-like glycans. And this has been in the case of not just of N-glycosylation, but more recently in these lower two papers, in the case of O-glycosylation as well. There's a pipeline of IgG monoclonal antibodies that have been worked on in plants. And of course, there's far too many to describe here, but I'll just mention a few that we are working on in my own lab. We have a particular interest in the broadly neutralizing antibodies that we heard about yesterday, and we've expressed a number of these. And what I want to show you here is some of those FC mutations, like the YTE mutation, or indeed the LS mutation, also work in plants and provide prolonged half-life in a mouse model. As Ellen was saying yesterday, we took on some of those Rabies monoclonal antibodies, and we showed that they were well produced in plants, and we also showed protection in a post-exposure prophylaxis model. And there are a number of other antibodies that we're particularly interested in, chikungunya, snake venom, and M. tuberculosis, to name just a few. So the capacity for producing monoclonal antibodies in plants is growing. And on this slide, I show the number of manufacturing facilities which are already established around the world. In red are those that came up very early on in the development of the technology in the 90s and the noughties. And of course, as you would expect, most of those have appeared in the global north, particularly in the United States, Europe, and in Japan. And you can see some of the products that these facilities are attempting to produce. In blue, though, are the newer facilities. And I think one of the important take-home messages from this is that we are starting to see manufacturing facilities developing in the global south. And in particular, I'll point out one in South Africa, there's one in Brazil, and although not in the global south, there's one in Bangkok, which I think is particularly important, as it demonstrates that final advantage I mentioned about plant biotechnology, that is of relative ease of technology transfer to low and middle income countries. So these manufacturing facilities look like? Well, some of them look like this, sort of, as you might imagine a greenhouse to look like. This is one that we work with in Germany, in Aachen. And here you can see transgenic tobacco plants growing as you would expect in a standard greenhouse. You can go up a level, of course, and this is a state-of-the-art plant growth facility using the latest technologies, including LED lighting and vertical farming. And this is one in Texas. But of course, you can go the other way and go, and this is the one that I mentioned, which has been developed in Bangkok and is now in phase 2 clinical trials with a SARS-CoV-2 vaccine, which has been regulated by the Thai FDA. And so you can see that you don't need these highly sophisticated manufacturing facilities. And in fact, this is a relatively inexpensive facility that was developed by this startup in Bangkok. So the field of molecular farming has really been through a developmental process. And I would say that the companies who were making the very first products, as you might expect, chose products which were the low-hanging fruit and provided the opportunity for rapid development paths, such as orphan drugs and animal vaccines and, in fact, cosmetic ingredients. More recently, though, there has been increasing interest in the high-value biologics, so we really are moving into the sort of the mainstream biologics world now in terms of IgG monoclonal antibodies and human vaccines. And what I just wanted to spend the last couple of minutes on is just thinking about where we might go in the next few years. And I think the next generation products really ask the question, well, what can plants do where plants actually provide a key advantage, not just in cost and scalability, but in other technical areas. And I think the areas that we would be looking at would be multi-component products, so cocktail products. And we heard yesterday also about the importance of antibody combinations in the case of HIV and rabies. We think there are some opportunities for recombinant proteins as alternatives to antibiotics. And a particular area which I think has been ignored in this meeting is complex antibodies like secretory IgAs. So very different antibodies for very different applications. So beyond IgG monoclonal antibodies, of course, I think there should be a lot of interest in the mucosal antibody, namely secretory IgA. And as you'll know, this is a much more complicated protein compared to IgG. And in particular, it's adapted for the mucosal environments, where, of course, we encounter most of our infectious agents. So secretory IgA is a decameric protein. That's why it's complicated. It's a complex of 10 different proteins. And it has high functional affinity because it is tetravalent. It has 4 antigen binding sites, essentially comprising 2 monomeric antibodies dimerized and then enwrapped in a 4th protein called secretory component. Now, these antibodies can do what IgG does. They can neutralize, they can aggregate, but they're also highly glycosylated, and that's one of the reasons they are resistant to proteolysis and adapted for the mucosal environment. And it has turned out that these are quite difficult to produce in CHO systems. In fact, secretory IgAs were first expressed in plants back in 1998. But more recently, we have identified major improvements in the productivity and the efficiency of assembly of this complex protein. And we recently reported our work against ETEC using a plant-derived secretory antibody and its effectiveness in a murine diarrhea model. And of course, SARS-CoV-2 has also not escaped our attention. Of course, it is a mucosal pathogen. And although the vaccines are important, I don't believe they prevent carriage or indeed mucosal transmission of the virus. And so in this case, I think passive immunization with a secretory IgA may well be advantageous. And we've worked with a number of monoclonal antibodies to SARS-CoV-2, and I just wanted to highlight one in particular. We start with IgGs and class switch them, of course. The IgG in this case was very poorly neutralizing, but when we made the secretory IgA secretory IgA1 version or the secretory IgA2 version, in fact, we enhanced the neutralization by a factor of about 10. And we think that this is largely down to the increased functional affinity or avidity of the secretory antibody. Of course, if you want to deliver antibodies by the mucosal route, you have to find a good way of doing it. And we've been looking at nebulization or aerosolization. We've identified a method to do this. And as you can see from these traces, the SIGA1 and the SIGA for 2 SARS-CoV-2 antibodies are resistant to degradation in this device, even though, of course, it elevates temperature and molecules. So we have found a way to stabilize secretory antibodies in this environment. And so we're quite excited about the possibility now of delivering secretory antibodies to the upper respiratory tract. So just to summarize, the field of molecular farming has matured. There are commercial products, although, as I said, these are the low-hanging fruit. There is a pipeline of products in development, including vaccines and antibodies. It's a suite of technologies, but the established transgenic and transient expression systems are probably the most important. And of course, the manufacturing facilities established around the world have led to increased regulatory understanding of the technologies. Where do we go and what are the upcoming challenges? Well, we've seen these A's before, the acceptability, accessibility, and affordability. And one of the key things then will be the correct product selection and in collaboration with players in LMICs. We need to think about regulatory acceptance of these new technologies in LMICs. We have to consider public acceptance in areas where GM in plants is still not generally accepted. And we need to work much harder on technology transfer of our manufacturing platform. Thank you very much. I'm very happy to take any questions.
Thank you, Julian. That was a fantastic overview. And I was actually really surprised to see that map that you showed of all of the molecular plant farming facilities around the world. So, I mean, the field clearly has progressed. And I think your point about IgA is well taken. I mean, we have focused on IgG, obviously, because all monoclonal antibodies, or most monoclonal antibodies, are produced on the CHO cell line. But certainly, you know, I think we shouldn't be ignoring that ways to produce IgA antibodies will be particularly important for mucosal pathogens. So I think that's a really, really good point that you made. There are some questions in the chat. One of them is about the regulatory opinion on this technology, which I think you addressed in your last slide. But the other one is about when do you think we will see maturity and large-scale industry manufacturing of this technology of plant production?
Well, I think we did earlier this year. Medicago, a Canadian company, produced a SARS-CoV-2 vaccine using transient expression technology in Nicotiana benthamiana, and it was approved by the Canadian FDA. So that was a joint venture with GlaxoSmithKline. I should also say that the first product of molecular farming, which was an enzyme called glucocerebrosidase, was developed by an Israeli company. But I think it was almost the day after it was licensed by the FDA, they were bought out by Pfizer. So I think the big players are well aware of what's going on.
All right, well, thank you very much. I'll hand it over to the next presenter. So we're going to hear from Moderna. So Obadiah is going to take us through mRNA coding for monoclonal antibodies. So the floor is yours.
Great, thank you, Erin. Just get this in full screen here.
Yep, looks good.
Okay. Great, laser pointer, we should be good. Thank you very much, Erin and Alexander, for the invitation to join this important discussion today. I'm happy to share, some human, mouse, and NHP data on mRNA antibodies, because I think it's informative on the current state of where mRNA technologies stand. Just for clarity, I am an employee of Moderna. Please don't trade on any information I share with you today. And over the next couple minutes, I'd like to give you some perspective on the mRNA platform in general. I'll highlight some of the work that has now been published, which had been presented on previously on the first mRNA antibody to go into human testing, mRNA 1944. Also walk through some data on biodistribution, local routes of administration, and combinations, which are areas of active research and areas where mRNA may have some complementary advantages to work in the recombinant space. And also I'd like to highlight some future directions for RNA-coded antibodies, some of which have been touched on by a number of the speakers already. So as we know, mRNA is kind of the middleman in making an antibody. It starts with DNA, which gets converted to mRNA and then translated into protein. And some of the properties of mRNA guide its usefulness in the infectious disease world for antibodies. It's a temporary set of instructions, allows a cell, in the case of humans, it allows B cells to make antibodies. And that transient nature of mRNA is kind of akin to a lot of infectious diseases, right? In the case of pandemics or evolving variants, you need a lot of versions of antibodies to be able to neutralize all these escape variants that are arising. So in some ways, mRNA is uniquely positioned to be a technology that can be used in the infectious disease space. And I should also say, the work I'll be talking about here today is really around Moderna's mRNA technology. There are a lot of other approaches, out there, using self-amplifying mRNA, using oRNA, eRNA, even other mRNA technologies, modified, unmodified nucleic acids. So we'll be, talking today about Moderna's technology. And whether it's an antibody or a vaccine or a therapeutic, there's a very, similar path that all mRNA-based medicines take, within our company, and that is it starts with a sequence, right? And that sequence can come from, in the case of an antibody, can come from a B cell from an infected individual. It could come from, some type of antibody discovery campaign. It can be an engineered sequence. In the end, it's a sequence that comes, in-house, and then it gets converted into an RNA sequence. That's obviously a fast process, takes, hours, minutes, on some level, and then it goes into manufacturing. So those first three steps from a sequence to getting into the manufacturing queue are relatively straightforward. We're looking at a period of days there. That manufacturing of an mRNA is somewhat different than what we've heard from a lot of the technologies that have been described thus far in the last 2 days, and that it's not a cell-based approach. It's an in vitro system where it starts with a plasmid is made that gets converted into linear DNA. There's an enzymatic process, IVT, which produces a purified RNA, which is shown, in the middle here. And that, in some cases, can be, a stopping point. It can be a storage point. Purified RNA, when it's stored in the freezer, it's a very stable molecule. But if you're interested in, moving quickly, then, that RNA gets formulated into an LNP, and this has been well documented throughout the COVID-19 pandemic, both from work, at Moderna and a number of other mRNA companies, that LNP formulated mRNA can be made. It can be made at clinical scale, it can be made at global scale now. And then it's stored in formulation there in 0.4 here. Then it's ready for either injection, IV infusion, whatever the point of use is. So in general, the process for making, when people think of mRNA, you often think it's a very fast process. It is, and that's largely because it does not involve, it does not involve mammalian cells, it doesn't involve cell line engineering, and it relies on the best properties of nucleic acids, which are their informational molecules, and they can be made in the same manner over and over again. And I will say, largely, depending on the size of molecules that one is working with, RNAs, have very similar behaviors based on the size of the molecule. So it's less about the sequence, more about the size in terms of their overall stability. So within a size range, there's a variety of sequences kind of very similar properties, which may or may not be the case for a lot of biologic proteins. We've heard about some of the challenges in the space around dealing with the proteins as APIs and having the proper controls in place to be able to concentrate them and use them in clinical formulations. So with that technology in place, again, going from a protein sequence to an RNA sequence, really our first approach here in advancing an RNA antibody into the clinic was with mRNA 1944. And this mRNA 1944 codes for the CHIKV24 antibody, which was discovered by the Crow lab at Vanderbilt a few years ago in collaboration with Moderna. And that antibody potently neutralizes chikungunya virus. It was really a great proof of concept system from our perspective, in that we knew it was an ultra potent antibody, potency down in the low nanomolar range for neutralizing the virus. And we had a benchmark in terms of the circulating serum levels of antibody that we felt would be protective. So this is the first antibody we chose to advance into the clinic. The technology that we're using at the time, which is largely what we use for a number of programs internally, is to code a heavy chain and a light chain on separate RNAs, co-formulate them into an LNP, and then deliver those intravenously. So in 2 RNAs, 1 LNP formulated product administered intravenously. And here in this cartoon, we kind of see on the bottom left over here, that LNP with 2 RNAs in it gets taken up by a cell. Those make their way to the ribosome, produce a protein, which then gets secreted into the serum. And then in the clinical studies, we're measuring serum protein levels and neutralization activity of those proteins. The phase 1 study was intentionally very conservative in the initial version, looking at 3 different dose levels with sentinel groups in each. And it was expanded based on the safety profile from that initial study to include repeat dosing, which I won't share the data here today, but that has been published, and also plus or minus the use of steroids as well. What we can say is that from looking at the lipid pharmacokinetics, there was no toxicity associated with the lipid. It was rapidly degraded as it was designed to be, and did not demonstrate any accumulation even after repeat dosing. And here, these are dosed kind of 1 week apart. We see very comparable decay kinetics here for the lipid. More interesting is the amount of protein that was able to be made from these groups. Again, 3 dose levels, 0.1 to 0.6 mg per kg. We saw what we were hoping to see in this case, which was dose-dependent increase in protein levels. So of all these, the different groups here, we saw good half-life from the antibody that was produced of 69 days. The only change that was made from the CHIKV-24 antibody, which was a wild-type antibody that we started with, was that it was converted to the LS FC modified version. So the half-life of the antibody was good, or on the high end, even for an LS modified antibody. And we did see this dose dependent increase in protein levels. And on the top right here is what we're really talking about. If you look at the y-axis, that's the serum concentration in micrograms per mil, and the x-axis is times. This goes out to basically a full year. In the larger box in the inset, I think it's also illustrative to highlight that at time 0, there's no antibody, right? You've administered RNA to a subject here, but over the course of 24 to 48 hours, that's when the protein is being translated and produced, and that's when it's able to make it out of its cells, and that's when you're able to detect it in serum. So that is part of the properties of mRNA. It's transient molecule, gets quickly converted into protein, and then that mRNA is degraded and does not produce any protein after that. So if you're thinking of an infectious disease where you need a lot of antibody in a couple of days, then RNA technology is a good fit for that. And also the one thing I will say is that these circulating serum levels of antibodies those are well within a therapeutic range for a number of potential applications of antibodies in the infectious disease space. One thing that was really of note from a lot of the COVID antibody work over the last few years is that the PROVENT study from Mark Esser and the team at AstraZeneca really showed that if you give a COVID antibody prophylactically and you're in that low microgram per mil range, it's completely as effective as vaccination. And that goes out to 6 months when you're dealing with some of these long half-life antibodies. So I think if you're kind of considering applications of mRNA, there are prophylactic applications. There are also a number of therapeutic ones. You could potentially make kind of intact IgGs in a relatively short period of time in vivo. And this work has been published in Nature Medicine last year. So that's kind of where we stand on the clinical side of things. As far as I know, that's the only RNA-coded antibody that has had a clinical readout. I know there are a number in development from a number of different companies and academic groups. But I also wanted to share with you today, some preclinical data that we have that looking at RNA-coded antibody combinations in collaboration with the Gates Foundation, and then also another Gates Foundation collaboration that we have. So the idea here in this HIV BNAB project was to take representative antibodies from 3 major classes and see if we could combine them together with RNA, knowing that there may be some engineering that's needed to be done there, because we were concerned with the technology that we work with, meaning heavy chain and light chain on separate transcripts, that there can be cross pairing and mispairing between heavy and light chain. So we took 3 representative classes of antibodies: CD4 binding site antibodies, V1, V2 apex antibodies, and glycan V3 antibodies, and took a step back to see if we could convert each of those first from fully intact IgGs, where there would be 2 RNAs per IgG, to single chain molecules with an FC on them, or scFVs. And that would allow us to use 1 RNA transcript where we're not concerned about having light chain pairing, just potential mispairing between heavy chains, which could actually in the end lead to some interesting biospecifics or bispecifics. So, long story short, I won't go through, all of the engineering that was done, but of the 3 major classes of antibodies, the V1, V2 apex antibody, PGDM 1400, and the glycan V3 antibodies, each converted well, going from heavy chain or going from fully intact IgGs into the single chain formats. The CD4 binding site antibodies did not, and that was consistent across 3BNC 117 and 6 and one of the NIH antibodies. So we opted to move forward with 2 single chain antibodies for V1, V2 apex, glycan V3, and then a fully intact IgG for the CD4 binding site. And that would allow you, to potentially produce, 3 different specificities. In vivo, heavy chain and light chain should, pair together nicely to form fully intact IgG on the left-hand side. And there can be some, like I said, bispecifics or monospecifics, which could be hybrid type molecules that could be produced. The idea was to take these molecules, co-formulate them, inject them into a mouse, and see if we could identify neutralization of specific clades associated with each one of these antibodies in vivo. And that's what was done here. We used an IV administered co-formulated antibodies. Again, N6 is a representative antibody that's a full IgG. The others, PGT121 and PGDM 1400, single chain versions. Those were co-administered to human transgenic FCR mice. And we monitored, or we basically pulled serum samples for the first 20 days and projected concentrations, out to 90 days. So the plot that runs out to 90 days, those are based on modeling data. And we looked at the neutralization potency of each of those samples against 3 different clades of viruses. And then we were able to back calculate the concentration that could potentially be, in each of these serum samples. And we did see what we were kind of looking for in the study, which was neutralization of 3 different clades of viruses when you're giving essentially 4 RNAs coding for 3 types of antibodies. And we were encouraged by this, and also encouraged by being able to hit that kind of 10 microgram per ml threshold out at 90 days with these particular antibodies, being early generations and not the most potent. That's kind of the ballpark that we had to hit from a concentration standpoint. So this was really the first time that we had shown this work, and this work has been submitted for publication here. And we'll keep folks posted on that process. So in general, some antibodies converted well from fully intact IgGs to single chains. We were able to successfully co-express those antibodies. We demonstrated that they're functional in independent neutralization assays against 3 different clades of virus. But there's a real need for HIV antibodies that have better properties, that have higher potency, and we're really excited about some of the collaboration work that's ongoing with the IAVI group around EPGT121 type molecules and some of the engineered versions that have much higher potency and higher breadth. Because with that combination, then you can get to antibodies where you don't have to go to such high combinations, and you don't have to go to such high concentrations either. To really get to important therapies. And the other thing that I'll say here is that there's, completely glossed over the amount of engineering work that's involved in going from an IgG to a single chain format. Some cases that was successful, some cases it wasn't, and also introduces some non-natural modifications into these antibodies. Actually, in all cases, they're non-natural. So really, starting from a technology where you're selecting for molecules that are in the format that you want to use, potentially like VHHs, that's what we're going to hear about in a couple of talks, might be one way to kind of take out that engineering component, which can kind of slow down the overall process. So starting from the right starting point of a molecular base to your technology, I think is important. So I also want to share some preclinical data, again, sticking with the HIV theme here, and further collaboration with researchers at the Gates Foundation around the PGT121 antibody. So again, the similar antibody to what we were talking about in the past, although this is the fully intact ALS modified PGT121. So it's an IgG. In this case, we wanted to look in NHPs, and we wanted to see first, can we make this molecule in NHPs? We had a feeling that that was going to be fine based on some of the previous work with mRNA 1944. So can we make it? What does the PK of that antibody look like? And then importantly, we wanted to start to look at alternative routes of administration, really getting to a locally administered antibody to see, really set a baseline for where our technology stands in this space. So 4 groups here. The first group in blue, the top group, is an IV IgG, so just PGT121, the recombinant protein. Then we have an IV mRNA version of the same, again, using the formulation that was used in the chikungunya virus vaccine. This is a clinical formulation. And then we had 2 IM groups where we administered mRNA with 2 different formulations. And we were very encouraged by this study. And if we first just look at the slopes of the curves here, the recombinant protein, half-life and decay kinetics are very similar to what we saw with either IV or IM administered mRNA, which was great to see. And also say the serum levels that we're seeing with the IV mRNA as well were a little bit above what we had anticipated based on the recombinant protein alone. It was also encouraging to see that the IM groups here, again, with multiple different formulations, did produce the same protein, again, with very similar pharmacokinetics at lower levels, even though there was the same dose. And that, we have seen that in some studies. We tend to see more protein being produced from IV administered mRNA versus intramuscular administered. But some of that is going to be case by case, depending on the antibody sequence. So this was, the first set of NHP data we had on these types of molecules. We wanted to take a closer look, not just at the serum levels, but also at the levels of protein that one are seeing in other compartments as well. And that's where I just want to highlight some biodistribution data here. And this is a consistent theme with a lot of LNP related technologies, are that on the left-hand side, again, this is a 24-hour time point of the exact same study we just talked about, where in blue is the protein group where it's given IV, and then the other 3 groups are the RNA groups, with the IV infusion group there in red, giving the highest serum levels in that 20 to 30 microgram per mil range. Again, that's just a 24-hour time point. But if we start to look in the lung, we see a difference there, and this is where IV infused antibodies, in this case, the PGT121 antibody, had about 5 times more antibody in the lung than one would have anticipated just based on the serum levels. And I think that's important. This could be, this is an N of 1 for this particular study for this antibody, but I do think it highlights, some of the differences that one might see with mRNA-coded antibodies. And that's important not just for HIV antibodies, but for developing mRNA antibodies on a global scale, or that you really need to know what you're making in vivo and where it's going, and taking a more detailed look at the biodistribution of those antibodies is important. And we'll come to that and kind of what I think are some of the questions and concerns, or open questions, maybe is the way to say it, in terms of the field going forward in the RNA-coded antibody space. So some of the challenges, again, I just highlighted understanding the biodistribution. I think it's going to be the biodistribution of an RNA-coded antibody is going to be different depending on the route of administration, and that needs to be studied very thoroughly, and that will play a role in how regulators view nucleic acid technologies, both in terms of what safety studies are required and aren't required. So just taking the old kind of playbook of how antibodies are developed, and then applying that to nucleic acid antibodies, I think it's not going to work. So I think the path forward there is understanding at a detailed level biodistribution. That's where we're doing a lot of work internally at Moderna, and in collaboration with a number of folks that are part of this network to help answer some of those questions. There's also a characterization question around in vivo expressed antibodies, and what I mean by in vivo express antibodies are that we really need to characterize what's being made in hosts from RNA. It was well documented, here over the last, couple of days that there's an incredible amount of characterization that's required in the recombinant antibody space. Now, some of which, can be, you can leverage a lot of history in terms of, different in vitro or cellular expression platforms, knowing that, we can control glycosylation in some ways, aggregation in other ways. But there's really not a huge body of data to go off of in terms of an in vivo express antibodies. Do we really know exactly what cell types are making these antibodies? Do we know exactly what the glycosylation profile is, what the biodistribution is? So in some ways, points 1 and 2 here are tied together, but those are areas, of consideration for deeper scientific understanding. And that all kind of ties into the path forward for regulators, and that, I think that, the group from Mass Biologics and, Mark and his team have highlighted, as leaders in the field, some of the requirements that, might be needed for bringing forward a recombinant protein. You have to do a tissue cross-reactivity study. Well, does that make sense to do with a nucleic acid technology? If you're making a recombinant protein and running a tissue cross-reactivity study, do you really need to do that if you're injecting mRNA and it's producing something that's slightly different in vivo, meaning from a glycosylation profile standpoint or from a post-translational modification standpoint? So I think the path forward with regulators highlight as one of the considerations that could impact accessibility, particularly in LMICs, because over the last couple of years, really mRNA medicines have just started to make their way across the world, and I think that's going to be an area where we really need to leverage scientific understanding of what we're making from mRNA to then use that in an educational way with regulators, while also being able to be a bridge in some levels to the recombinant world that is kind of thriving out there for antibodies for infectious disease. So with that, I would like to acknowledge a number of the folks that have been part of this work, and really look forward to sharing with you some exciting data that will be coming over the next couple of months in the antibody space, based on a lot of these collaborations. In particular, the U.S. government agencies, DARPA, the NIH, and BARDA have been just fantastic collaborators and supporters of nucleic acid technologies, not just Moderna's, but in the field in general. And I should absolutely point out the work that Devin Salk and his team have done at IAVI, and the great collaborators that team has been with us on a number of projects, some of which are highlighted here. And also the interest that the Bill and Melinda Gates Foundation has had in this work has been impactful, and is one of the reasons that we've done a lot of work, starting with the HIV antibodies, and now that will be branching off into other ID pathogens. So I should also highlight a few folks, at Moderna in particular. Andrea Carpio is our head of research, who's given us and myself a lot of freedom on a scientific level to explore RNA antibodies. Sunny Himatsu and Kaylin Deal are leading and have led a lot of our antibody work, and our whole development team, which was able to pull off a fairly complex, CHIKV antibody study here a couple of years ago, just pre-COVID. And also there's, a shameless plug for a review that Kaylin authored here on the bottom in Vaccines on RNA-coded Antibodies. I think it has a broader assessment of the RNA space, even beyond Moderna's technologies. And with that, I'd like to thank everyone here for giving me a few minutes of your time.
Thank you. That was excellent. A lot of questions were popping up, but then I think on your final slide, you addressed quite a few of them, including around biodistribution, and some of the sort of the regulatory concerns. I had a question about whether or not, if you increase the dose given intramuscularly, because the doses were quite small in any case, so you could envision giving more via an IM route. Do you think if you increase the dose, you would see the same expression as when you give the intravenous infusion?
Yeah, so I think there's an upper limit in dose, to be honest. And that's really just based on the concentration of RNA that one's dealing with. If it's formulated, say at 2 mg per mil, and there's a volume limitation that you have for IM. I think sub-Q is probably similar, but there might be more flexibility there in terms of how much one could give. And, there may be differences in terms of, the amount of protein that one can get out of a localized, administration. And we're going to just find that out experimentally to see what the limitations are there.
And the other question I have before I move to some of the questions in the Q&A is around thermostability. And I'm assuming that it's the same as mRNA vaccines. At the moment, they're sort of, sensitive to temperatures outside of ultra cold chain. They can go into
absolutely
for a certain amount of time.
Yeah, and while the LMPs used in these studies are different from the LMPs used in clinical studies, so we don't know as much, as we do from the COVID vaccine world. We do kind of treat them the same way, so treat them with cold chain storage, and that's obviously an active area of research in the field to get to, RNA or nucleic acid technologies that are more thermostable.
All right, fantastic. A question on cost. You're at early stages of development, but are you able to sort of give any kind of range in what the costs might be predicted, cost of goods for mRNA coding mAbs in terms of based on the chikungunya monoclonal?
Yeah, I wish I could. I mean, the back of the envelope calculation that, one can use, which is probably wrong in the end, but if you look at, 100 microgram dose of a vaccine, and we know what those cost, the doses that were given in the Chicks B study, 1944, are in the milligram range, right? So it's not in the microgram range. So we're looking at, maybe 10-fold higher amounts or more. So there's kind of a balancing act between what we're seeing in the world, which is, nucleic acid technology, manufacturing capabilities are scaling, in real time. So I think what a vaccine or a RNA cost today will be very different even a year from now. But we're about 10+ fold off from a dose standpoint. So I think you look at it as there's a real chance that these are in the same prices as vaccines here in the next couple of years.
Okay, great. I also have an interesting question on co-expression of accessory molecules. So sort of example chaperones. Would they enhance antibody expression? Have you looked at that?
So I think that's a really interesting area, and I was fascinated by Julian's talk just beforehand, as well, and specifically in the IgA space. I think how molecules are made in B cells is going to be, or how antibodies are made. There are specific chaperones that help them get out of cells. I think that's an active area of research. So I think that is one area where we could find step-fold changes in terms of how much we can train other cell types to make antibodies with nucleic acids.
Right. Well, I see there's some other questions in the chat, but we are running a little bit behind actually. So maybe I'll take note of some of those other questions, and I might shoot you off an email. And if you have time to address any of them, I could contact the people that asked them. So thank you very much. Super interesting. The next presentation is we're going from mRNA to yeast. And Chris Love from MIT is going to present on yeast expression. So Chris, are you on the line? Oh, yes, you are. And I see that your presentation is coming up.
Right, thank you. And it's a pleasure to be here and participate in this conversation about what the future of manufacturing could look like. What I'd like to do is take the next 10 minutes plus whatever questions you might have to take a step back and to really talk about why we are going to have to embrace new technologies, including the biology, in order to make affordable monoclonal antibodies as proteins, and to make accessible manufacturing technologies that could be available in many parts of the world to improve robustness and response. So a good MIT talk always has an equation, and I'll start with a very simple one. And I think this is actually really important that we keep in mind as we address the questions of cost and scale and production. If I want to make anything, including monoclonal antibodies, there's sort of certain levers or factors that we have to pay attention to in building the processes and ultimately the economics for manufacturing. We've touched on these in a number of conversations, but it's helpful to put them in context here. We always need more. And more doesn't necessarily mean more total product. It means more yield. It's more product for the materials and input that are required in order to generate that material. It should be faster. The more throughput we have through the facility or the space volume that we use for production will ultimately translate into lower cost. And the more efficient we run that space has a direct impact on the overall cost of goods. And we would want better. And better in this case means really less deviation from whatever specifications we've put on the product that we're attempting to make. We should reject less, and we should be able to improve the overall productivity through better attributes and controls of that final product. And that these sum together in some form with some equations in between get to lower costs per unit produced. What's important here is that this is agnostic and does not address at all cells, cell-free, mRNA, modalities, how we do this. This is really just about what's required in order to evaluate a technology to incorporate into manufacturing changes. And so what I think we can learn from this industry and from others is that holistic changes in the prevailing technologies or the scales that we operate at are often needed to realize these step changes in costs that are associated with the targets that we might be looking towards here. And so it's with this in mind that I think we should think about the rest of the conversation, both here in my talk and I think in our discussion time later, as to what's going to be really truly necessary in order to realize the goals of affordable and accessible monoclonal antibodies. And so what I'd like to do is kind of walk through this a little bit more and kind of unpack a bit the biology that's required in order to realize these changes. And we've touched on it in a number of different talks already today, but I think it's important to remember that biology is the common denominator for making biologic medicines. It is intrinsically invoked in every step and part of the process of producing a final good. It impacts the amounts that we make, how efficiently we make it, the time that we make it in, and the costs that go into that. And so the choice of your biology here will impact all of those costs incurred and the time in order to produce the final material. And I think the thing that's often overlooked in thinking about this decision or the conversation around what is the host biology is the rest of the process that it fits into. The host biology itself shapes the complexity and ultimately the costs of the process that's necessary in order to realize the production. That is, that the biology is only one piece of the total equation that's necessary in order to reach an affordable monoclonal antibody, yet it has a large influence on how we design and operate that process. We've already heard today in Mark's talk about the C1 system, how a fast host can lead to high productivities on a daily basis. Looking at this in comparison and thinking about those types of technologies in relation to other existing technology platforms, you can see the choice of the host influences the number of operations that I have to undertake. And I've only shown here the ones that are value add processes, things that change things. It has nothing to do with how many times you have to exchange medias or operate steps in between the ones that are shown here, as well as some of the estimated times to go from the point that I crack the vial to the time that I have final formulated bulk drug substance in this example. So putting that in context with the first equation, the more raw materials and labor required, the more operational steps. These things are proportional to one another. So from a optimization standpoint, I would like fewer steps. To go faster, I want to process all that same bulk of material in less time. That is that my grams per liter per day per meter cubed of facility used should be optimized to be as large as possible. And through talks like the one from Just and Evotech earlier, we hear examples of how modular manufacturing can help facilitate this by consolidating the operating space. And then ultimately, better quality can come from things like automation and continuous production, as there's plenty of examples in the field now to do this. And the question is given those hardware technology implementations, what's the biology that would help support this? And to address the question that the folks have raised in a few of the other talks here, it was a nice study done by Ellen Coleman, a master's student at MIT in conjunction with Amgen, that looked at what would be necessary to reach comparable cost of goods of making monoclonal antibodies in CHO today with yeast. And it's about 50% of the titers that you achieve in a CHO system. And this comes back to something that Mark highlighted in his talk on the dyadic system, is the speed with which these types of organisms can operate. So that sounds great, but what biology or biocatalyst? Remember, a cell is just a collection of biochemical reactions that transform raw materials into final goods. What's the biology that's going to transform this? Well, in order to take advantage of the advances of what we're learning in the hardware engineering side of facilities, we need a tractable genome that's easy to edit so we can program it as we like. Should be fast and flexible for development. Again, there's drug development costs that we could consider outside of the actual manufacturing here that could be addressed. Extracellular expression is critical. This is really the key difference between microbial production of biopharmaceuticals today and mammalian systems, is that most of those products are made intracellularly, which requires additional steps of recovery. So extracellular expression, as we see in CHO cells, is an essential aspect of what's needed for biology going forward. And of course, quality is part none. It is the thing that we must maintain in all aspects here. But it's actually not something that's intrinsic to the host if designed properly and the target product profiles match the requirements. And so the good news here is that there's lots of solutions. We don't need to talk about one host or one solution to the challenges here, but there's lots of biologies that are probably sufficient for these features given the appropriate development. And as has already been highlighted, there's clear examples in other industries where low-cost protein production is required to make commodity goods that are priced at a point that are well below what we typically see for production of mAbs today. And so, with that in mind, how should we start thinking about this as engineers? Well, alternative hosts now really have become a lot like software. Our ability to read the genomes through sequencing technologies lets us precisely know the locations in the organism, both where native genes are expressed and how, but also where in the genome you may be able to put new chaperones, new genes of interest, new heterologous proteins that you want to express in various ways in a safe way inside that host to maintain stability and maximize productivity. And then, of course, with advent of tools like CRISPR-Cas9, the ability to write into that genome to afford those changes is now straightforward and can be adapted to specific hosts based on its own host biology. And so in combination, we can now cycle and design these aspects for the features that we want. And the question is what traits would we necessarily want in an ideal host to make monoclonal antibodies for infectious diseases? Well, I think we understand we need to make these at large volumes and at low costs and with high potency. So what are the features of the biocatalyst that might help facilitate that? We know what the end goal is, but what's the thing in the middle? Well, the very practical way to think about this is, again, as a biocatalyst, we need a very high conversion rate of the input feeds, carbon, nitrogen, and oxygen, fundamental elements here, to make a complex chemical on the other end, a monoclonal antibody. Good news is that fermentation science understands this exceptionally well in a number of other areas where the conversion rates of carbon sources and feeds into this are known and can be described with precision. This is something we probably need to come back to in thinking about how we actually focus on improvement of cellular hosts towards the objective of making monoclonal antibodies. And we've already heard a little bit today also in Mark's talk on the dyadic system, but thinking broadly about the implications for process engineering of the rest of the solution around the host. Reducing susceptibility in genomic variation or contamination is important. Improving the purity of the reactor fluid coming out and facilitating new recovery approaches, perhaps crystallization or precipitation would be essential. And then compatibility of these hosts with perfusion-based technologies for continuous manufacturing and or very large scale fermentation would transform those scales in accessibility and in affordability. So to this end, in 2018, we formed the Alternative Host Research Consortium at MIT. This is a pre-competitive collaboration to enhance microbial hosts. I've listed here some of the objectives we have, thinking around Comagatella fafai, or Pichia as it's known, as a first example of how to think about engineering hosts for production of these. Some of our current members are listed here. And we've adopted an open source approach to this research consortia to really build an ecosystem that's focused on how to realize the full transformation that's necessary to pull these hosts into the mainstream. And I won't have time today to unpack all of the advances that we've made in this space, but just simply to highlight a couple of things. We've been working towards improving IgG1s. We now have batch productivities of these that are comparable to CHO cells in optimized CHO-K1 cell lines in batch. This is, I think, again, highlighting some of the speed. You can see we're reaching these productivities in 3 to 4 times faster than what's reported for these same cell lines in CHO. We've been able to make a number of different monoclonal antibodies in aglycosylated forms, including ones that are relevant for SARS-CoV-2. Many of these show the similar kinds of binding to the targets as what Mark had showed for the C1 system earlier. The question in PICIA is always about glycoengineering. We've revisited this, taking a slightly different approach than what's been described previously. The pathways for this are really well understood, but the challenges in some of these hosts have been described is that they have poor viability or growth characteristics in fermenters. So we've gone back and asked questions about how the host natively regulates glycosylation machinery and used that to inform engineering strategies. So here's examples of where we've taken the kind of canonical approach, and you can see lots of higher mannose structures evident in relation to the targets. But as we modulate these gene expressions in the appropriate ways using native promoter sequences, we can get to uniform G0 constructs on reporter peptides. So I'll finish with a couple of thoughts so that we have time for questions. There's some implications here that I think we can dig into more. Lowering the cost of manufacturing of mAbs will require holistic technology changes, both of the biology and the hardware solutions in order to accommodate this. We can certainly dig into the limitations on where CHO can get to in pricing, but these are well documented in the literature. I think we have to rethink that 10x kind of change that's necessary and what's required here. I would posit that the host biology itself is a very accessible and substantial lever for engineering improvements towards that goal, and we need to think more about that. And to that end, catalytic funding that's invested now in pre-competitive research on these host biologies is really what's needed in order to realize the impact in the next 5 to 10 years because of the regulations in this space. And this is probably important for both medicines like we're talking about now, but also alternative foods that are going to similarly require complex proteins to be made in high volumes at commodity prices. I think the other half of this equation is the product itself, and we've talked a bit about this, but I would suggest that the manufacturability is something that must be considered at day one of thinking about any R&D project on a target of interest. Fundamental biology is important to understand what's necessary for neutralization, for example, but actually developing a product that can be manufactured at very large scale may require thinking about other factors like aglycosylation or FC engineering to elicit the right effector functions, alternative proteins like the LMICs or designed binders that could replicate similar features of the biology, but in a more scalable production technology. And then finally, I think actually is really important is thinking about reducing the barriers to entry to participate in manufacturing. And this is something that can be enabled by technology changes in both the host and the hardware that could expand innovation of products and ultimately enhance global robustness in responses to emerging diseases. Thank you. I'll stop here and be happy to take a question or two if there's some time.
Thank you very much, Chris. That was excellent and very clear. And I mean, it sounds like you're doing great work in this consortium. There is a question in the chat about, apart from yeast, what in your opinion is the most promising host biology production of monoclonals?
Yeah, I think it's a great question. Some work that was done funded by the Gates Foundation that was a collaboration between Biogen and Amyris a few years ago that I had an opportunity to participate with, did a head-to-head comparison of different host biologies. And I think from that, diatoms, leishmania, fungi, yeast, a number of things were considered, not to optimize them fully to the best of their abilities, but to really understand what's it like to work with. And I think from that, yeast and fungi are probably the two most promising. They're also the ones that have the largest industrial enzyme experience. And so there's a prior knowledge elsewhere in the community on how to make proteins in this, obviously for different purposes and sort of to different quality specifications. But I think that just in terms of tractability, these are ones that exist today.
Great. Well, thank you very much. And you mentioned VHHs, which is a good segue to our next presentation, which is from Laura Crowell from Sunflower Therapeutics. Laura, you're there, I see. Thank you, Chris.
Can you see my slides?
So we can. I think I've got to change my view though.
Yes, I can.
Yeah, I can see.
Okay, great. Well, thanks everybody for sticking around. I'm going to change gears just a little bit here and talk about nanobodies or VHHs. So we'll start with the question, what is a nanobody? Nanobodies are derived from the variable region of heavy chain only antibodies that can be found in species like camelids. They have some unique properties. They're very small, so they're about 12 to 15 kilodaltons typically, and they have high stability, solubility, and potency. Because of some of these properties, they offer a significant opportunity as products. There are multiple potential administration routes for this type of protein, including the potential for oral delivery. There is regulatory experience for these proteins as well. The first VHH-based therapy was approved in 2018, and they can be specific to a very wide variety of targets, including unconventional targets that are difficult for larger antibodies to actually reach. So taking a look specifically at infectious diseases, there are a variety of VHHs that have been identified for infectious disease and tested at a variety of non-clinical stages. You can see here just a quick summary, but we have nanobodies for things like cholera, norovirus, rotavirus, and even some in development for COVID. So the reason why VHHs are really interesting, in particular when we're talking about lowering cost and increasing accessibility in low and middle income countries, is that these types of molecules are highly manufacturable in simple hosts. So this draws right off of Chris's presentation and a lot of the other things that we've heard today about looking at different biologies to actually produce our products of interest. So I'll talk a little bit about Pichia pastoris as an example here, but a lot of these advantages could be true in other simple hosts as well. So nanobodies have small size and limited post-translational modifications, and this can lead to high titers in simple hosts. So for example, there's some literature on unoptimized processes for Pichia showing greater than 1 gram per liter of VHH production in fed batch. These molecules can also be purified without the use of affinity resins. We all know that protein A is a very expensive step, but it's critical in the production of mAbs. With these nanobodies, there have actually been processes developed that do not require protein A and also avoid things like His tags, which can't be used for regulatory concerns in commercial manufacturing. So process experts at Sunflower have actually developed a platform process for the production and purification of VHHs using the host Pichia pastoris. On the production side, we utilize perfusion fermentation. So we have proprietary in-vessel cell retention device that lets us do perfusion with Pichia. And as we've heard with these kind of alternative hosts, you can start making your protein very quickly. So in this case, in under 48 hours, we're able to start making our protein after inoculation. And our protein is secreted with a very high initial purity, actually up to 80%, which simplifies the downstream processing. So in some unoptimized processes using Pichia to create nanobodies in perfusion fermentation, we see titers around, volumetric productivity around 0.3 grams per liter per day. To put this in perspective, this is roughly equivalent to a 4 gram per liter titer in a CHO fed batch process. And again, this is unoptimized. So there's a lot of room for improvement here. On the downstream side, our platform process uses a two-step non-affinity purification process. So we use 2 ion exchange columns, and that is able to reduce the host cell protein and DNA below non-clinical limits. As was previously mentioned, we don't have to do things like viral inactivation, so that removes those kinds of steps from our manufacturing process. When you don't use an affinity-based chromatography method, it's a little bit harder to have a platform process because typically the way that your protein interacts with an ion exchange column might depend greatly on the protein itself. What we found in particular for nanobodies, because they have a conserved structure, is that there's actually really one spot in the chromatography that differs, and that's in the elution conditions. And you can predict the best elution conditions based on the protein sequence itself. The platform process that we've developed here is also compatible with straight-through chromatography. I won't go into that too much, but what it means is that you can eliminate things like hold steps and buffer exchanges between your chromatography columns, which ultimately allows for more integrated and automated manufacturing. So we have actually used this platform process to develop and create nanobodies and show that it is both safe and the process development is very fast. So on the left here, I'm showing just a little bit of data on a VHH that is specific to influenza that was tested in mice. So this VHH is called E13. We tested the molecule created in E. coli and purified using a His tag as compared to the same molecule produced using our platform process in Pichia. So you can see based on the body weight of the mice over time after infection with influenza, both the Pichia-based and the E. coli-based molecules were able to maintain body weight and also significantly reduce the titer in the lungs. Some benefits of this platform process are repeatability and speed. So we have successfully produced and purified 8 out of the 9 attempted nanobodies that we have tried to do. And these are specific to a variety of targets, including rotavirus, norovirus, and influenza. And I think maybe even more impressive, using this platform process, we've been able to go from a protein sequence to non-clinical material in as few as 5 weeks. So I talked a little bit about this platform process. And one of the really important things about it is that it's actually deployable on next generation manufacturing equipment. So we've heard a little bit today about, what do we have to do to manufacturing to actually make these products more accessible in low and middle income countries. At Sunflower, we think the solution here is small footprint and automated systems that could allow for in-country or in-region manufacturing. And we've actually developed 3 different scales of system for this purpose. I'll note that the data that I showed you earlier was all generated on a precursor to these systems. So all the way on the left of this slide, we have here our Daisy Petal system. This is a benchtop bioreactor system that enables perfusion fermentation of microbes. In the middle is our end-to-end benchtop solution, the Daisy system. This system includes modules for perfusion fermentation, for purification using straight-through chromatography, for formulation using tangential flow filtration, and a refrigerated collection fridge. So using the Daisy system, you can go from your inoculum of cells to fully formulated bulk drug substance in a collection fridge. The entire piece of equipment is integrated and automated. So a user walks up, connects their fluids, puts in their cells, loads a recipe and hits go, comes back a couple days later to formulated bulk drug substance in the fridge. Our Dahlia system is a larger scale version of the Daisy system. We call it kind of pilot scale. It has all of the same modules and is similarly automated. And we estimate that this system can produce about 10 kilograms of protein output annually. The system takes up less than 50 meters squared. So to put that in perspective, that is less than 4 parking spots. So you can consider that the facilities needed to deploy a system like this are much, much smaller than typical manufacturing facilities. Now, unfortunately, I don't have data on the production of VHHs in these specific systems yet, but I do want to highlight a bit of data on a similar size molecule using these specific manufacturing systems. So we were able to produce a protein subunit vaccine candidate for SARS-CoV-2 based on the receptor binding domain. And one thing that I want to highlight here is the high quality protein was made in a very simple manufacturing environment. So we deployed the process on our DAISY system, actually in a carpeted office building, and we were able to produce thousands of dose equivalents in less than a week. We had purity and safety near our targets, and importantly, in all of our samples tested, we saw no bioburden. Similarly, on the larger scale Dahlia system, we deployed this manufacturing process in a completely bare warehouse and saw the same quality attributes. So I think this is really promising data that efficient manufacturing systems can actually produce high quality protein in very simple environments. So if we think about combining the platform process for VHHs with this unique manufacturing approach, we can actually really imagine in-market manufacturing for VHHs. So with the platform process, you can do rapid manufacturing development for a variety of different VHH candidates. And then using the small footprint automated systems, you can enable regional manufacturing. We've heard a little bit about this so far over the last 2 days, but I just want to point out a couple of the key impacts that in-region manufacturing of biologics really can lead to. The first one is improved product accessibility in low and middle income countries. We've talked a lot about cost of goods and price, but Sunflower spent a lot of time in the past few years talking to stakeholders in low and middle income countries. And a lot of times they just have no access to the proteins that they're trying to get. And they actually have some money, like they're willing to pay for the products, but they just can't get to them at all. So having this accessibility is really, really important in addition to driving down the cost. In-region manufacturing can also lead to sustainable businesses within the region. This can create ecosystems that can support both the research, development, and manufacturing of products. And ultimately, this can lead to the production of regional products for regional markets. The VHHs that are needed in one place in the world might be slightly different than what's needed in another place. And these ecosystems can actually enable for site-specific development of products. So with that, I'll just acknowledge the Sunflower team. We are a unique women-owned and led business, and we're quite proud of that. And you can see we are a very small startup, only 7 full-time employees right now. So very proud of everyone for establishing these manufacturing systems and showing the potential for local manufacturing. With that, I'm happy to take any questions.
Thank you very much, Laura. To be perfectly honest, I have not been following the antibodies space. So I think it's actually really cool. And I think if you can get rid of protein A, I mean, you can reduce your costs significantly. I see that there's a question in the chat. So how is GMP achieved and maintained using the automated manufacturing systems?
So right now, our manufacturing systems have not been deployed in a GMP environment. We have designed them with GMP capability in mind, and we are currently undergoing another design revision to reach commercially ready systems, and those will be GMP compliant.
Fantastic. And I think your point is about, local production is really important. And I think now more than ever, there's going to be a push for countries to have their own production, not only of vaccines, but for these high-cost biotherapeutic products, because it not only improves their access in the long term, but It also, as you said, improves the just general economics in those countries. So fantastic presentation. We're actually running a little bit behind, about 20 minutes or something. What I would suggest is we just take a quick 5-minute break, and then we'll come back to the presentation from Bill and Melinda Gates Foundation. So if we could come back at 40 past the hour. Thank you, everybody. Just a quick break for everybody to get some coffee, including myself. Thank you. The next presenter is Jacqueline Kirschner from the Bill and Melinda Gates Foundation, who's going to talk about some of the efforts that they're doing at the foundation to advance biologics for global health, infectious diseases. Over to you, please, Jacqueline.
Thank you so much, Erin. I just would like to say thank you for the opportunity to tell you about our efforts in biologics and alternative hosts at the foundation. And just wanted to say how much I've been enjoying the presentations today and really impressed by the breadth of expertise on the call. So hopefully we'll have a great discussion at the end. Before I begin, I just want to give credit to Laura Shackleton, a Senior Program Officer at the Foundation, who I work closely with on these biologics efforts, who's currently on leave, and Laura Powell, who's a consultant who's backfilling for Laura Shackleton while she's on leave in what I call the Laura Exchange Program. And also Steve Hadley, who's also on the call, who has led the efforts of the foundation for really trying to reduce cost of manufacturing from conventional CHO cells, as well as exploring alternative hosts, and is able to address any questions regarding those efforts. And I'm trying to advance now. Okay, so I think the foundation is largely known for vaccines, and we certainly do a lot of work in vaccines to prevent disease for a number of different global health indications. But what might not be as well known is that in the last few years, we've really ramped up efforts in biologics in a number of different global health indications, including malaria in the pneumonia space, respiratory syncytial virus, COVID for the last 2.5+ years, HIV. And in all of these cases, we're trying to prevent infection and or prevent disease. So in general, it's the same use case as we have for vaccines, but a completely different approach. And we know that antibodies are the fastest growing drug class. We know they're very safe, but they are largely unaffordable for low and middle income countries. And we think that that kind of inequity really needs to be corrected. And so we do have a number of efforts in conventional monoclonal antibodies that I will tell you about. And of course, just to remind everybody, we've heard about this already a bit during the talks yesterday and today, but the conventional monoclonal antibody process is typically, what we have done is to use humanized mice that express the human immunoglobulin repertoire, largely through collaborations with Kymab, which is now part of Sanofi, and or to screen for monoclonal antibodies from human convalescent donors or humans who have been vaccinated and have a great antibody response to that vaccine. And these sources have been tried and tested, and there's many different ways to sort for antibodies from either one of these sources. But once you get these very potent antibodies, then really the issues come into play downstream, which is how do you make them as low cost as possible? And so with investments early on in Just Bio, before they were acquired by Evotec, investments were made to really try to reduce the overall cost of manufacturing of antibodies, but also really to optimize the monoclonal antibody sequences to make them much more developable. Manufacturing is one part of that developability profile, but many other attributes are equally important for making sure that an antibody is well behaved and leading to the lowest possible cost per dose, including things like reducing aggregation, improving pharmacokinetics, and really just making sure that it's very fit for this particular platform. Often that means making variant sequences that could affect the activity of the antibody, and so they have to be evaluated carefully. And this whole process is quite important for reducing the overall cost of antibodies, but it does take more time. And that is an approach that we have used for some conventional monoclonal antibodies that have come out of the discovery efforts, two of which have transitioned to the Gates Medical Research Institute. which is a product development partner of the Gates Foundation that we work very closely with. One antibody is for RSV. This is called RSMO1. This came out of our collaboration with Arsanis. It was discovered actually at Adimab some years ago, but variants of that were made through the collaboration with Arsanis and partners at Just. That has just completed a phase 1 study at the Gates Medical Research Institute. Like some of the other RS3 antibodies we've heard earlier about from Ruth, this does target the prefusion F protein. It's site 0, as she mentioned. And it is a very potent antibody. We are still looking for more potent antibodies in order to try to bring that cost of goods down even further. And there's 2 efforts ongoing right now. For malaria, we have an antibody that was discovered through a pipeline of different sources of lead candidates coming from different monoclonal antibody discovery efforts that went through standardized assays. And out of that work came one particular lead candidate called MAMO1, which was licensed by the Gates Medical Research Institute from Atraca. This targets, like the very exciting antibodies that Bob spoke about yesterday, Cis43LS and L9LS. This antibody also targets the circumsporozoite protein of the malaria sporozoite in order to prevent infection of hepatocytes. And while we are very, very excited about the clinical data that Bob has described and will be getting published soon in the next 3 or 4 weeks that he from the Mali studies that he's worked on together with Pete Crompton, who I believe is also on the call. In the modeling we've done, it appears that one of the major drivers to bring the cost of goods down for both the RSV antibody and the malaria antibody is really the pediatric dosing. So, tiny humans need tiny doses, and that allows us to bring the cost of goods down to something that can meet the TPP. And in our TPPs, we're generally talking about $10 per dose or much less, ideally more like vaccine-like pricing. So it's a pretty aggressive goal to try to get antibodies to fit into that box. And it's not easy, and doing conventional CHO cell manufacturing may not be sufficient to get us there with the current technologies. So it's really exciting to hear about all the different technologies on the call yesterday and today that may help us to get closer to that TPP. We also have had an effort on COVID monoclonal antibodies for the last 2.5+ years. And ideally, the initial goal was to find a cocktail of monoclonal antibodies that would be resistant to escape and could be made affordably for LMICs. That effort has now pivoted to something that we didn't think we would have to say, which is a pan-SARS-CoV-2 antibody, but also really thinking about how to get to a pan-sarbecavirus monoclonal antibody-based product and/or maybe even a anti-beta coronavirus product for future pandemic threats. So while we've had these efforts in conventional monoclonal antibodies, as mentioned in many other talks and reiterated here on this slide, there's a lot of drawbacks to conventional monoclonal antibodies. First and foremostly are the high cost of goods. This really does preclude the use in LMICs or limits the use in LMICs to certain populations. And it really prevents us from having the enormity or the full impact that we'd like to have to address the global health problems in infectious disease. We've already talked about how high doses would require IV administration, and this is also difficult to do in remote settings and rural settings if you're outside of community hospitals. So this becomes something that is really untenable for us to meet the TPP for most use cases and for most global health indications. We've talked about the short half-life. Antibodies can they have a longer half-life than, say, a number of small molecules, but their short half-life compared to the vaccine response that you can get. Cold chain dependence is an issue for some of the rural and remote settings, and it includes, and contributes to the overall cost of delivery. And then we've talked a lot about, in COVID times, escape. This is something that I think happened much more rapidly than anybody had predicted. It's become a huge issue. And so addressing it by having a cocktail of antibodies, of course, increases the cost of such a product. So that's not necessarily a great solution. And antibodies sometimes have insufficient breadth, as we've learned about from HIV BNAb. So again, these problems with monoclonals and their great specificity can be a drawback for the cost of certain products where you need to have multiple specificities in order to address the global health problem. So listed on the right here are a number of potential solutions and gives you some insight into what we consider end-to-end strategy for how do we address these issues for monoclonal antibodies. And it's not just about making a better monoclonal antibody or a cheaper monoclonal antibody. It's really everything from increasing the potency of biologics, exploring alternative hosts for manufacturing, not only for monoclonals, but for some alternative biologics, which I'll talk to you about. Exploring alternative routes of administration. So for respiratory disease, we're looking at intranasal and inhalation routes of administration to see whether we reduce the cost per dose by using a dose that actually gets right where you need it to act, rather than giving a systemic dose, a fraction of which actually biodistributes to the target tissue. We're looking at alternative delivery methods. You've heard the great talk from Obadiah Moderna about mRNA. We're also looking at circular RNA, which is a very new RNA delivery method that I would say is at the nascent stages, but potentially very promising. So all of those first solutions with, I'd say, increasing potency, probably the biggest lever, could all help address the high cost of goods problem. In addition to address the short half-life, alternative delivery methods may help with that as well. If we can get long-lived delivery vectors or long-expressing delivery vectors. We've heard about Fc modifications to optimize binding to FcRn. These are half-life extension mutations. The most common ones that are clinically de-risked are YTE and LS, but there's many other ones that have been identified academically that have not yet been clinically de-risked and might be of great interest to further understand their utility. We're also looking at slow release formulations, which can further extend the half-life by allowing the depot of monoclonal antibody or other biologic to be administered subcutaneously and then enter into the bloodstream through Fickian diffusion. It may also even have the added advantage of increasing the thermostability. In terms of cold chain dependence, again, these novel formulations may help with that. But there's other biologics that we're investing in that are inherently much more thermostable. We've heard about VHHs, but there's other computationally designed or modular proteins that we think could address this issue. And then finally, this idea of escape and insufficient breadth. We have a number of investments in different multi-specific modalities that can address this issue and also potentially be more potent to drive down the cost of goods per dose. So what are these things we're talking about? So on the top row here is a human monoclonal antibody, conventional antibody, IgG. We know and love this. There's over 150 approved products that are human IgGs or humanized IgGs, and they are great, but they have this high cost of goods. So what can we do better? So for a multimeric alternative to an IgG, we're looking at both secretory IgA and IgM antibodies, which, as we know, will have much higher avidity for the target and therefore could have higher potency. But they do require CHO cell manufacturing, although we're exploring some alternative hosts for secretory IgA. And the COGS may be higher than they are for IgG. That could potentially be offset by much higher potency if we can figure out how to improve the half-life. They are known to have much shorter half-life than IgG, and there's work going on at IGM Bio to engineer IgM molecules to have better half-life and better biodistribution. So that's of great interest. Another multi-specific, multimeric technology that we're investing in is multibodies. And I'll talk to you in detail a little bit more about what these are, but these are ferritin-based, human ferritin-based multivalent structures that display up to 32 moieties, a combination of Fc and Fabs. We've seen neutralization potency for viruses increased in vitro, 100 to 1,000-fold, sometimes 10,000-fold, depending on the starting IgG potency. And they are being engineered, the Fcs are being engineered to enable IgG-like PK. These also require CHO cell manufacturing. It's not yet clear what the cost of goods might be. CMC issues might be. Those are things that are ongoing areas of research. And there's no toxicology data on these yet, but they've been administered to animals and do not have any overt adverse effects. So in addition to these antibody-based or antibody multimeric structures, we are looking at some other alternative biologics. You've heard about nanobodies already, the VHH domain molecules. Advantages of these are that they can be produced in E. coli or yeast, which allows maybe small footprint manufacturing, possibly cheaper manufacturing, and certainly faster manufacturing. They're highly thermostable, so that could be cold chain disruptive. They may have better biodistribution to certain target tissues, which might be useful for some of the global health indications that we're interested in solving. But they do have a short half-life unless they have some domain added to them to mediate a longer half-life, such as an FC or something that binds to a long-lived serum protein. Neutralization is the only mechanism of action, so you lose out on some of the advantages of an antibody, which can have effector function that's mediated by the FC. And some of these might require a device, which may or may not price them out of the TPP, cost of goods criteria for administration. So just some considerations there. Another alternative biologic that we're looking at, you've heard about DARPins already. We don't have a particular investment in DARPins right now, but we're very interested in this technology. It came up in COVID times. This is a Molecular Partners asset that was then acquired by Novartis. And they did have clinical data with DARPins for COVID, which was quite impressive, showing about a 2-week half-life. These are modular ankyrin repeat-based proteins. That's where the ARP comes from. And you can have different modules that are linked together that can target different epitopes within a particular protein target, and or have a modular domain within the DARPin that can bind to a long-lived serum protein to confer that longer half-life. We also have an investment in the Institute for Protein Design mini protein technology, which is similar to DARPins in that these are small modular proteins that are linked together, but these are computationally designed. So it's completely in silico design of proteins based on a known structure. And those computational methods, as they improve, might allow for very rapid response to new pandemic threats. Both of these can be made in E. coli or Pichia. They have a short half-life, and so similar to the VHH nanobody approach, they have some drawbacks, and unless they have some domain that can mediate a longer half-life, as I've just described. And finally, there's a peptide lipid conjugate investment we have that is interesting because this can have broad specificity for whole virus classes. This can be administered intranasally and potentially can have efficacy even in the lungs. We don't yet know what the COGS might be. Again, short half-life would necessitate use of a device for delivery and probably frequent delivery. Okay, so just to give you a quick bit of detail on, so much time I have, on the multibody. So this is published work. This is coming from Jean-Philippe Julien at Sick Kids in Toronto, with a lot of work from Arif Jeta, who works with him closely. And again, these are ferritin-based structures that can express both Fabs and FCs on the surface. And it's the multivalency of that Fab expression that confers high avidity to the target proteins. You can also have up to 3 different Fab specificities, which will allow you to take something like an IgG cocktail, which is shown here in a COVID SARS-CoV-2 virus neutralization assay, comparing the combination of cocktail of 3 different IgG molecules to those same Fabs put on a multibody, and you can see that the potency is dramatically increased in vitro. We are trying to understand the PK-PD relationship for these molecules in vivo, and whether the potency increases that are observed in vitro will translate to significant increases in potency in vivo. So those studies are ongoing, but it seems very exciting, very promising. And again, in addition to COVID, there's published work also on an HIV multibody. Just going to show that quickly here. The reference is there. But just to make the point that in addition to this increase in neutralization potency, you can see that the multibodies also provide a significant increase in breadth, allowing a single biological entity to provide not only the potency and resistance to protect against a panel of HIV viruses. And the one other, on the other end of the spectrum, is this kind of computationally designed mini protein that I mentioned. Again, there's publications on this work shown here at the bottom of the slide. They had two different approaches for COVID, which was to take structures that look like ACE2 and find proteins that mimic ACE2 that could bind to the SARS-CoV-2 spike receptor binding domain. And then they also tried to find things which, based on, again, protein structure and computational design, could block the interaction between ACE2 and RBD and bind directly to RBD. So those two different approaches were taken. They had a number of different candidates that came out of that work that were potently binding to the target. Again, these are very small proteins. They're linked together as modules in order to confer the avidity. Highly thermostable, again, can be made in E. coli or Pichia. And we are in a collaboration right now with IAVI to explore the utility of these in the clinic, hopefully within about a year's time. And I just want to mention very quickly, since this is supposed to be a conference on manufacturing, I just told you a lot about our approaches for end-to-end thinking on alternative biologics, alternative hosts, alternative routes of administration. The alternative host investments are listed here. Some of these you've heard about already from other speakers, but just to mention that we are looking at all different kinds of hosts that we think are useful for manufacturing and expression of monoclonal antibodies, but also some of these alternative biologics where monoclonal antibodies may not be suited to that particular host. I think the ideal is to have a full monoclonal antibody that has the post-translational modifications that allow it to function and behave like a human monoclonal antibody that's made in CHO cells. But in parallel, I think that there's different use cases where small proteins could be made very quickly in a pandemic situation, or can be made in a small footprint. And so looking at other expression systems such as a marine protist, Pichia, and even optimizing Pichia for better manufacturing of an FC nanobody fusion protein is of great interest. And then finally, we're even looking far afoot to spirulina for production of VHH nanobodies and single chain FV molecules. And I will stop there and take any questions. Thank you so much for your time.
Thank you so much, Jacqueline. I'll actually, in the interest of time, table questions for the panel discussion because you're going to be sitting on that later.
Sure.
So I'll hand it straight over to Maria Teresa from FDA to talk about some of the manufacturing experience that they have on the regulatory side of things.
Hello, can you hear me and can you see my screen?
We can, thank you.
Wonderful. So my name is Tere Gutierrez. I don't know if you can see my camera. Let me see if I can share. Okay, I can do it without camera. So my name is Tere Gutierrez. I am here with my colleagues Joanna Su and Stephanie Troy, representing the FDA. And I'd like to take the next few minutes to share a manufacturing perspective on the production of these monoclonal antibodies for infectious diseases. So the usual disclaimer, please refer to FDA guidance and specific in the relevant review team for specific regulatory issues. Okay, so I think I'd like to start by indicating that the general CMC development considerations and principles are applicable to all biological products, including monoclonal antibodies developed for infectious diseases. And we heard in the last 2 days how critical is to define the intended product, the quality target product profile, and consider aspects such as the intended population, the disease, the dose regimen, the route of administration, and other considerations that are needed to manufacture a product that is intended for use. And of course, this quality target product profile or the desired product characteristics should drive the product design, the development, and optimization. We heard about some strategies about potentially extending the shelf life or modifying the potency of the product, or formulation development approaches to make these products more suitable for intended use. We also we heard some talks on the potential to apply relevant monoclonal antibody platform knowledge, and these platform approaches can be used throughout the development of these products. Screening, for example, from the lead selection by the use of screening platforms, or using platform knowledge for the development of manufacturing process and the control strategies and formulation. I think we can usually think about the manufacture of a monoclonal antibody following a particular steps that are usually well defined as a standard manufacturing process for a monoclonal antibody. Of course, there will be certain considerations for certain products, but in general, I think there is a big opportunity to take advantage of that prior knowledge and platform knowledge. And we also heard about some strategies that could increase the yield of these products by using high yield expression system of other modern technologies that could facilitate production and making these products more accessible. So, but of course, we also are aware that, one of the hurdles for manufacturers are the regulatory considerations, right? What is needed for, with respect to CMC to open an IND, what data are needed to support development of the product and for licensure. Again, here I would say that the general expectations for monoclonal antibodies are the same regardless of the indication, with product-specific considerations. And one thing that we usually we like to encourage is early communication with the FDA to align CMC development with regulatory expectations in clinical development. Sometimes what we have seen for certain products is that the CMC development may be lagging, and then there may be too many gaps to fill when the product is leading to licensure. And another thing that I would like to share with you is, the possibility to explore regulatory pathways to accelerate development of these products. In this slide, I try to summarize some of the approaches to accelerate product availability or approval that are available at the FDA. One thing is that I would really encourage manufacturers to start early and frequent communication or consultation with the FDA through different types of meetings, and you can find more details about this type of meetings in FDA guidance. But we have seen that this type of communication facilitates development and also clarifies potential misunderstandings and could reduce the number of review cycles for a particular product. Approaches that we have used for making these products available for emergency use are the expanded access INDs. And of course, all of us are very aware of the use of emergency use authorization, which obviously this is a temporary approach. This is not an approach that will be used for a more continuous availability of the product, as these products will be available only during emergency. Other programs that you can think of or you can consider are fast track or breakthrough therapy. And with respect to approvals or licensure, you could consider accelerated approval, the use of animal rule or the priority review. And given that we have limited time at this this conference, I would refer you to FDA guidance. There is a link here for the guidance where you can find more information on these programs. And of course, direct you to our website for any guidance that you may need for your development of your product. Obviously, not only CMC, but other aspects of product development. This was a very short presentation, but we wanted to take the opportunity to talk to you, to talk in this forum. The email addresses, my email address is here, and also of my colleague Joanna, who we are both representing CMC. And with this, I would like to thank you for your attention. I could potentially take questions now, or maybe we can leave them for the panel discussion. Thank you very much.
Thank you very much. I think you did really well to summarize everything that you're doing at FDA and the various pathways you have in a very short amount of time. So thank you. We actually only have about 20 minutes left. So that's 20 minutes for the panel discussion. Unfortunately, I'll just display just what the question was and who we were going to ask to present on the panel. Unfortunately, Umesh from the Serum Institute of India, who's the director of R&D there, has to catch a flight. So he's unfortunately had to drop off the session. But I think we can still, just I guess what we can get out of this session is just sort of a bit of a summary from some of the panelists about what they're doing. And we might not have time to get to the FDA questions, but thank you so much for offering your emails to any participants on the call that might have any direct questions. So I'll start by handing it over to Eric Palfren. He works in the European Medicines Agency, specifically in the Office of Vaccines and Therapeutics. And just like, similar to what the FDA just covered, is we'd be really interested to know about, the types of approval pathways at EMA that are available, and also about any challenges that you see from the regulatory perspective and some of these products.
Yeah, thank you, Erin. This is very interesting. And I've also listened to the, the presentation for the FDA, and we complementary with their, what they have given as possible pathways, and so we are more or less on the same line, I would say. And it would be very difficult for me to go in very specifics, and I can only give high-level kind of support that we offer. As you know, obviously, as a regulatory authority, we are gatekeeper, and we look for safe, effective medicines of high quality. But our second function is also that we enable research, and we have discussions with applicants, with industry, either on an informal basis or more formal procedures. And so, this is not only obviously for monoclonal antibodies, but more generally, one of the most important, and it's not a surprise, is that companies can come for scientific advice, by which we look at the different areas of quality, non-clinical questions, the clinical issues can be discussed, methodological issues for development. Although this advice is not binding, it is nevertheless very useful, and it has also shown regulatory success in the future. Now, for monoclonal antibodies, if you consider this as an important innovation fulfilling unmet medical need, there is also a possibility to go into an earlier scheme, what we call the priority medicine scheme, by which companies can apply for, and if this is agreed, then they receive scientific advice at a much earlier stage, at the level of proof of principle and so on. And so it's a guided process with different interactions and early appointment of our rapporteurs, and multidisciplinary from the EMA people being involved, and so guiding the company in what tests has to be fulfilled. Another advantage of this so-called prime scheme is that the company can also then, once they have all the data at hand and are ready to apply for a marketing authorization, and that shortened review times apply, so an accelerated review from our side. Having said that, obviously, I mean, you don't need a prime scheme for an accelerated review. That can also be outside that when it's a product of major public health interest, and especially from view of the fact that it fulfills, it's really a therapeutic innovation which it offers. And so that's a major kind of things which I want to convey. We also have an interest specifically for monoclonal antibodies for quality by design, by which this is obviously pertinent to the quality aspects. and by which it aims to meet predefined product characteristics early on, so so-called enhanced approach procedure for quality, we call it. And it helps getting a better product and facilitates also the life cycle management in case changes are introduced later on. So the agency can also assist in this regard, and guidance is available on our website regarding the Prime Quality Toolbox for this quality by design. Then if I just take a few more moments, I know that you have phrased the question, but if I can just go and enlarge it a bit from the boundary and see on the excess of pharmaceuticals, especially with reference to low and middle income countries. And I know that obviously registration is one step. You have others, I mean, like pricing, distribution, the possibility of R&D, manufacturing and technology transfer to low and middle income countries. But nevertheless, I mean, it is as such that registration is a very important step, a first step. And here, I mean, the EU license could also be used by other jurisdictions. I mean, and for monoclonal antibodies targeted against infectious diseases. You know, when in the development program, different areas as well, low middle income countries were involved with participants recruited from these areas, and it is a disease of a global kind of interest, then our license we are giving can always be used as a blueprint for other jurisdictions to come to their own conclusions in their own licensing process. And that is interesting. And to finalize on this as well, we also have a special procedure, our Medicines for All procedure, which is also called the Article 58. And that is important because this is together with the World Health Organization that our scientific committee reaches a scientific opinion on the benefit risk of use of new products for places where it's going to be used outside Europe, and is mainly applicable for infections. And this is a process which in its assessment involves independent people, independent experts from the World Health Organization. And also we are in close contact then as well with the regulators in low middle income countries who will be involved and where the product is actually going to be licensed. On itself, the Article 58, the Medicines for All, is not a license, it's a scientific opinion, but it allows a very fast entry in WHO pre-qualification, and thus as well further for funding decisions to be made for bulk purchases by, for instance, UN agencies. One major example I would give on this is, for instance, the monoclonal antibody in prevention setting for malaria. As you can see that an EU licensing will have a different indication and would not be applicable for endemic regions. We would specifically have something prevention in the traveler going to the endemic country, while obviously in endemic country, the indication is completely different. And there an Article 58 scientific opinion could be useful. And this is more or less the major kind of information I would want to convey today to this meeting. Thank you.
Thank you very much, Eric. And I know you mentioned the prime eligibility, or maybe I'm saying that wrong, but yeah, I understand that nirsevimab, the first RSV long-acting monoclonal antibody, has qualified for that and is under that fast track regulatory review, which is great. And of course, the Article 58 with EMA has been absolutely crucial in giving a positive scientific opinion on the RTSS malaria vaccine, which is now pre-qualified finally. So thank you so much. In the interest of time, I'm going to jump to the next person shown on that list. So Umesh, I mentioned, had to drop off to catch a flight, but we would love to hear from Pete Gardner from the Wellcome Trust about some of the areas that the Wellcome Trust is investing in monoclonals for infectious diseases, and perhaps Pete, you could also say a little bit about any funding opportunities that might open up in the future for some of those on the call.
Yeah, thank you. Thanks, Erin. And it's great to be part of this meeting and see everybody on the call. I mean, it's been a fantastic couple of days. So for the sake of time, I'll try and whiz through and I'll give a bit of context. Welcome. We've had a recent sort of strategy refresh. And in terms of our infectious disease team, we're very much geared towards those most at risk and being better protected against current infectious diseases, but also the threat of emerging diseases and those that may escalate now in the future. And I think we're really thinking about how we can change the paradigm from sort of responding to intervening earlier to prevent that escalation of infectious disease. And core to that is the development of affordable medicines and equitable access. It's a critical part of that. And I think what I want to highlight is that antibodies for infectious disease, which is the area that I'm focused in, is really embedded in Wellcome's strategy, in the infectious disease strategy, and particularly embedded in our structure as well. And I think we appreciate that, we need a toolbox of medical countermeasures. So within the infectious disease team, we're not only focusing on vaccines, small molecules, but also antibodies. We have a growing portfolio. I think, there's a mix of ways that Wellcome funds. I think we're largely building on a foundation from the last couple of years of work done in our previous Enterix portfolio with my former colleague Lindsay Keir, who's been on the call, and also my Wellcome colleague Sally Nicholas. And so I'll go through a few of our current investments, but we also, as many people probably know, fund CARB-X and fund GHIT and fund CEPI as well. And there's some gathering momentum around monoclonal antibodies for infectious disease across all of those partners. And I think we keep a close eye on some of the investments made there. So for example, CARB-X have, 3 MAB assets for bacterial infections, progressing. And one particular of interest at GHIT is a MAB for malaria, which was funded by Wellcome directly at the sort of hit to lead stage, and that's now being funded by GHIT. That's a GSK monoclonal antibody for malaria, and that's in the sort of lead optimization stage. So we're delighted to see further GHIT funding for that asset. I think in terms of the current portfolio, it's largely split between enabling activities and sort of MAB discovery and product development. I think I wouldn't want to overlook the fact that we've been able to support WHO with putting on this meeting, and I think it's a critical meeting to pull together stakeholders to really discuss some of these issues and look at the challenges, but also the innovation that can tackle those challenges. I think so we're delighted that we can and provide some support to WHO. And I think there's a couple of other things going on as part of that package. So we heard yesterday about diphtheria. I think we're hoping that as part of the WHO Market Information for Access to Vaccines project, that there's going to be an assessment of EDAT to sort of better understand how MABs can plug the shortfall, the demand shortfall for diphtheria. We're also hoping to support the Rabies regulatory guidelines supplement that was discussed by Richard yesterday as part of the WHO norms and standards. So a couple of things there to really enable the environment for MABs for low and middle income countries, thinking about target product profiles and how to sort of lower barriers to investment. I think the other thing we've done is support IARVI together with WHO on the recent release of the preferred product characteristics of MABs for HIV prevention, and that's a great outcome. I think that's that document can really support the field to better understand how MABs can be used and deployed in low and middle income countries, and really getting the perspective of the end user feeding in early to product development, which ties into that angle of really, how do we deal with access? We've got to embed it early on in product development. In terms of MAB discovery and development projects, I mentioned our former funding of GSK's CSP targeting MAB for malaria, now funded by GHIT. I think we've got a couple of projects and focused on discovery of MABs for Shigella and Salmonella, and that's largely from our prior Enterix portfolio focus. So we're funding IARVI, and we're working with Toscana Life Sciences Foundation and GSK on 2 of those projects. We're also funding Savare GMP, not-for-profit group in the US, to develop and manufacture a low-cost MAB for cholera. And as part of that is some further optimization of CHO cell production. And we've heard a little bit from other groups on the call about how hard can you press current technologies, current CHO industry standard technologies to get an affordable product. And we also have funding to University of Aberdeen to develop a broadly protective monoclonal antibody for Candida, and that's in the lead up stage. I think so that's our current portfolio. It's a bit smaller than the Gates portfolio, but I think we appreciate the need to kind of focus on a number of different areas to take on those challenges. And I think very quickly, Erin, if I can just talk about sort of forward looking. I mean, I think we're really keen, particularly with this meeting, to really engage with the community to understand what are the priorities. And I think it was raised yesterday about the need to define a research agenda to support sort of coordinated investment for affordable monoclonal antibodies for use in low and middle income countries. And I think that's something that, together with other partners, other funders on the call, we're really keen to engage to do. I think three main areas of focus for us. I think we really want to support R&D for antibody scale and affordability. We've heard, it's obvious about some of the challenges there, and we want to sort of, invest in novel platforms. I think we know that we need diversity of platforms, and that there maybe isn't a one size fits all, particularly when you're thinking about tailoring to different pathogen needs and different use cases. I think we've got our eye on looking at supporting gene delivery approaches for antibodies, bioprocess innovation, and low cost antibody platforms. I think we want to sort of try and support and develop the development and testing of a select few antibody products. I think we're thinking about how we can support a product that could trailblaze access in LMICs, and we've heard a little bit about some of those some of those products might be maybe in the RSV space, the malaria space, etc. And I think we want to continue supporting activities to try and define that pathway of MABs to low and middle income countries. And I think we're hoping to expand very much on the work that we've done with IAVI, on continuing having conversations. I think we're hoping very much that as part of that, they will be facilitating a market forum around discussing novel business models to support sustainable and affordable monoclonal antibody access. So I think we're really looking to try and engage and build on some of the momentum that's been reached with other partners, like the work that Gates Foundation have done. But I hope that kind of gives a little bit of an insight into what we've done so far and what we're thinking about doing in the future.
Yeah, definitely. Thanks so much, Kate. And I think the role that the groups like Wellcome Trust and Bill and Melinda Gates Foundation have is just being able to provide that catalyst funding that sometimes it's just needed to get started. There is a question in the chat, and it's about the problem of suitability and access to monoclonal antibodies in low and middle income countries. What is Wellcome requiring in terms of monoclonal antibody access of its funders? So I guess this is a little bit like how the Gates Foundation has that global access clause in any of their funding agreements that they grant. Have you got similar language when you issue awards?
Yeah, absolutely, that's right. I mean, it's a critical factor, and thank you for raising it. So yeah, so we embed in our funding terms, in our standard terms, we have access requirements. But I think particularly when we're looking at funding into product development and for-profit companies, we put put in bespoke access obligations there, and some of those are driven by the pressures from UK charitable law. As a charitable funder in the UK, we have some pressures there. But we have to ensure that our funding can deliver on the charitable benefit and the public benefit of that. So absolutely, very much aligned with the Gates Foundation global access requirements in that regard.
Great, thank you so much, Pete. We're actually just about at time. We did hear just from BMGF, and we did hear from the CMC group at FDA. So I might take you guys off the hook for the panel discussion, but we have got somebody from FDA that's more on the sort of the clinical side of things. And so I do just have one question for Stephanie Troy, if you're still with us. And it's just about the animal rule, which is something that we heard about yesterday. In what circumstances can the animal rule be followed I understand that it can be used when the normal efficacy pathway is difficult to demonstrate. But what are the sort of general requirements, and for what kind of pathogens can it be used? And if you have an example, just so that folks on the call can have sort of a bit of an idea about that.
Thank you, Erin, and thanks for inviting me to this panel and this important discussion. As you mentioned, the animal rule is one pathway that can be used when clinical efficacy studies are not feasible or ethical. And one of the best examples I have was for the development of TPOXX for smallpox, because clearly we can't do a clinical efficacy study to look at efficacy with the smallpox, since it's no longer infecting people right now. There are several requirements, and these are highlighted in our guidance for industry about the animal rule, but mostly these are to ensure that the results from the animal efficacy studies could translate to people in terms of finding an appropriate dose, and in terms of the endpoints being translatable. And again, these are all highlighted in our guidance for industry. And I just want to echo what Terry said before, which is if anyone is considering this, we highly encourage you to contact FDA early and utilize our pre-IND program to discuss this. Thank you.
Thank you very much, Stephanie. There is one final question in the Q&A that's just popped up, and it's sort of just saying that there's been quite a focus on antiviral antibodies, which is actually, yeah, quite true, with the, I guess, the exception of malaria and diphtheria. But what's the panel's views on the potential challenges need for antibacterial antibodies? Does anybody want to take that? Maybe we haven't got the right panelists.
I'd be happy to contribute something. So yeah, no, thanks for the question. And sorry if I didn't mention that we did have some efforts for antibacterial monoclonal antibodies for prevention of neonatal sepsis. And there's been a lot of epidemiological work to identify the key pathogens that are responsible for neonatal sepsis in different geographies. The challenge, of course, is that there are many different serotypes. And so again, it becomes problematic to think about what would that monoclonal antibody product look like if you need to have that multiple specificity. So I think that's something that we felt was cost prohibitive at one point, but now that new technologies are coming online, it's something that we are definitely thinking about again. And like the Wellcome Trust, we do contribute to CARB-X, and I sit on a number of review panels there. So it's really interesting to see the progress that's being made for monoclonals for bacterial pathogens. I think it's very challenging, but absolutely worth the R&D investment.
Great. Well, thank you very much for stepping in there. We've gone a little bit over. I would really like to encourage anybody that didn't get their questions answered or has further thoughts or comments just to please send me an email. Thank you so much to the presenters and the panelists over the last 2 days. I think we had some really interesting presentations, and it's so nice to see that this field is advancing and that, it's looking like there may be more affordable monoclonal antibodies or antibody-like products on the horizon. So thank you very much again. Again, if you have any comments, questions, whatever, just please send them to me. We did mention that we will be sending out a recording. We do just need to check with a few of the presenters if they're okay with that. We haven't got confirmation from everybody. That might take us a day or two to get that confirmation. And if somebody doesn't wish to share their slides, then what we'll have to do is just cut out that section from the recording, which will just take a little bit of additional time. But certainly by the end of the week, we should be able to send you the recording and presentations that we have that we've got, what's the word, that's been authorized for us to share. All right. Well, thank you very much, everybody.
Thank you, Erin.
Thank you.
Thanks, Erin.
Thank you. Bye-bye.